Yeah, can we start actually?
No, sir. Just let's wait for 5 minutes.
Okay, okay. I may need little extra time, few
Oh,
minutes. Okay,
okay. Just you can. We'll be managing your talks, and no, that's no need to worry. Just let's
okay,
wait.
okay.
Also, some European countries as well as some Asian
Okay.
countries, they are not joining in proper time. That's why we need to manage the time.
Yeah, that's fine.
Yes, sir.
That,
I'd say for US people, it's early in the morning. That's why.
my audibility is little less. I'll be chatting in case I'm not able to hear you people. Okay.
Yes, we wish each one of them good morning, good afternoon, as well as good evening to all the participants. Myself, Mr. Bhupati, the representing Innovan Conference, welcomes all distinguished guests, dear doctors, scientists, educators, colleagues, friends, ladies and gentlemen. It's a great honor for me to welcome you to join this webinar on behalf of fourth virtual conference on cancer science and oncology that is started yesterday September 27th as 28 day 2 of the conference. I would like to express my most sincere gratitude for your presence in this opening ceremony for day 2 as a great way to initiate our scientific program. So today we'll be completing day 2 of the scientific program. Once again, I am especially thankful to Mr. Henry Bruce, Miss Nadia, who has her heavy burdens, duties, and responsible to make this happen today. I would also like to thank participants, especially those who are joining from a mile distance and sharing your valuable experience and ideas. With a good number of participants, I am sure this conference will be memorable, highly educational, and not to be missed event. The webinar aims to provide us aid for people to share analysis and results about all features of cancer science. This Virtual Cancer Summit 2021 will be covering the topics like cancer biology and genetics, organ-defined cancers, oncology subspecialties, radiation oncology, surgical oncology, precision cancer medicine and oncology, cancer biomarkers, cancer epidemiology, radiology, cancer preventive and research, cancer therapy and treatment, cancer pharmacology, cancer vaccines, cancer immunology and immunotherapy, oncology nursing and cancer, hematology, nuclear oncology, DNA damage, mutation and cancer, advance in cancer research and treatment, cancer pathology and genetics, cancer screening, and we have more than 45 to 50 tracks are there. Will be tracks as well as subtracks. We'll be covering some few of them among these listed what I mentioned. So make sure, I request all the participants, make sure to introduce everyone at the beginning. Ensure that you have clear, work appropriate background. Look into the camera when talking instead of looking at yourself. Be aware of your audio as well as video settings. If you have any questions or comments, you can text here so that we can ask behalf of the participants. So now I request Dave, one of the keynote speaker, that is Dr. Lakshmipati Vadlakonda, Professor of Cell Biology, Department of Zoology from Kakatiya University, India. The topic will be paradoxical role of lactate, pyruvate and pyruvate kinases in the cell respiration, metabolism and cancers in a historical perspective. Sir, I request you to start sharing your slide.
Thank you. Thank you very much. Good morning, everybody. And it is a pleasure to participate. Yesterday it was, I think you have disabled the participant screen sharing. Can we activate it? Yeah, now it's. Are you able to see this? Are you able to see this?
Yes, sir.
Yeah. Well, well, I am retired. In 2006, I retired from Kakatiya University. Afterwards, I was there in several institutes. It was actually as a consultant scientist in Cellworks, Bangalore. I realized that everything that goes on in the form of signaling and other things is only a peripheral system that needs actually, we need to understand the manifestation of these signal pathways in terms of molecular as well as the physical phenotypes. There's an old saying, this is present day model of cancer, an old saying, each one carrying a particular thing considers that it is important. There is an old cartoon from the Indian, this thing. Well, this is a cartoon which I have presented in 2016 in a conference, Molecular Science Society. Dr. Bhattacharya is the chairman for this, I believe. The title was Lactic Acid, a tiny molecule that sucks your money, muscles, and keeps scientists engaged for 2.5 centuries without telling what it is doing in the body. And today, several publications, particularly in June 2021, on nutrition, immunity, and global pandemic, gave us several answers for this question. I was examining this particular question for past 10 years in a detailed manner. I try to share some of these things and this thing. This is the model, current model, which we know, which we have been reading right from 8th class onwards, if I am not wrong. Okay. Now, this particular pathway, the triose phosphates and hexose phosphates are separated by one particular enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. That is where the aberration is also coming into picture. We need to understand that particular thing is my focus. Now, in this model, there's a reverse flux of actually the pyruvate to glucose has three loops. One is at pyruvate getting converted to phosphoenol pyruvate, classically called it as pyruvate recycling. Is it pyruvate recycling or phosphate recycling is a question which we are addressing. The second important is this particular enzyme is a highly controversial enzyme. The forward flux is a highly questionable point. Then, of course, these hexophosphate shuttles. One thing which we miss here is the glycogen's role. Glycogen actually contributes to glucose 6-phosphate in response to the stress response. We will see that as we progress into this. In my March 10th seminar in this particular conferences, I told you that there are two models of pyruvate entry into the mitochondria and two models of citrate synthesis, which raise several questions on the present models. One common attribute which we find that, whenever you find a deviation, you say the canonical mutation, blah, 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 like that, experiencing. So I went into the origin of this thing. These two were the background for which actually Krebs and Johnson took and then formulated this equation. This equation is nothing but a mixture of actually citrate synthesis and citrate breakdown in the cytoplasm. I can understand the problem there because they were working on the isolated muscles, not on the partition between mitochondria and these things. Ochawa group between 1945 and 65 worked on reductive carboxylation and condensing enzymes, and then they have stated that 80% of the citrate synthesis takes place because of the reductive carboxylation. That is what actually Martius group also have identified, and Johnson in 1939 confirmed that aconitase reaction favors citrate synthesis. Surprisingly, Krebs also has identified there are two models of citrate synthesis, but the first citrate synthesis is removed and then subsequent citrate synthesis will be stabilized in this system. That is what happens in G1 phase and G2 phase of the cell cycle. We'll come to this thing. Krebs adopted. Peculiarly, Krebs, although he started working with pyruvate, addition increased succinate. He was not confident of saying that pyruvate is the precursor of acetyl CoA. He has only stated that some unknown carbon atom, 3-carbon metabolite, contributes to the acetyl CoA. And today we have two amino acids contributing differently in G1 phase and G2 phase. 2019, I think you et al. In BMC Cancer, they have published an article where cysteine catabolism in the mitochondria contributes to the pyruvate, dehydrogenase, and acetyl CoA production into this thing. That's an important point that to be noted. And then there are several reports over about 10, 11 years, starting from Thomson's laboratory, Debrow's laboratories, etc., where reductive carboxylation of 2-oxoglutarate, alpha-ketoglutarate, it is also called, that actually contributes to these things. And now canvas is very large. I try to actually simplify as simple as possible. Now, the architects of modern cancer metabolism particularly is Warburg. There is no single paper which appears on metabolism of cancers without mentioning actually Warburg as a hallmark of cancer. Well, what are the deficiencies in this particular thing? Why we are not able to actually develop correct therapies was my first question when I started probing into this thing. In fact, in 2009, before Hanahan and Weinberg introduced metabolism into hallmarks of cancer, I addressed this particular problem of Warburg's in an informal lecture in my university. Well, Warburg proposed two models. One is posture effect, the second one is aerobic glycolysis. There are contradictions within these two proposals. In the first one, he says that oxygen inhibits glycolysis. In the second one, he says that oxygen produces lactic acid because mitochondria are dysfunctional. Well, these are two contradictions, self-explanatory contradictions in that. But Warburg's contribution to actually metabolic pathways comes more from his discovery from DPN and TPN, which are thermostable cofactors. That is where actually yeast alcohol production, both by posture and also Harden and Young, come into the picture. Okay, I'll come to that little later. Let us examine what Pasteur said. What is the Pasteur's data? Pasteur introduced the concept of aerobic life and anaerobic life, and in anaerobic life, he stated that yeast consumes 60 grams per gram of yeast and then produce alcohol. But whereas when lactic acid, this is very particular important statement, lactic acid contamination suspends alcohol production, and then phosphate and nitrogen in the media are transformed into macromolecule like albuminoid like material. Oxygen reduces the consumption of glucose to 4 grams to 1 gram of yeast. So 1 gram of growing yeast consumes 4 grams of this thing. If you take the ratio of 4 versus 60, it is 1:15. If you take the cytoplasmic ATP production according to the present models, which itself is an error, is 2:30. That is 1:15. So energy-wise, there is not much of a difference in these things. I now come to this thing. Pasteur's work was also a starting point for enzymology. Buchner introduced the term, I believe, zymase, and then at the end of this thing, he has also stated that cell-free extracts can also produce if this enzyme is present in that. Harden and Young extrapolated that work in the first decade of 20th century. In an extensive survey, they have identified that a thermostable cofactor and a phosphate that ferments sugar to alcohol. If phosphate is depleting or thermostable cofactors are depleting, then alcohol production will automatically get arrested. Warburg's credit goes to discovery of two thermostable cofactors. In fact, in 1935, he discovered this TPN or NADP and DPN was discovered, which we call it now as NAD in 1938. Warburg forays into metabolism ironically started with iron sulfur clusters, and iron he has labeled it as respiratory pigment. He won his Nobel Prize also for that particular contribution. And in 1920s, Meyerhoff introduced, in the next slides I'll come to that, Meyerhoff introduced the concept that glycogen depletion and lactic acid accumulation during muscle contraction provides the energy for muscle contraction. Simultaneously, the Core's couple introduced the concept of Cori cycle. There is a primary defect in this cycle because liver universally in any organism will not be producing LDHB, which is required for lactate conversion to pyruvate. Corey, in a later statement, stated that glycogen hydrolysis, because of the epinephrine activation, will be contributing to glucose production in this thing. That is, it hydrolyzes glycogen and then supplies glucose into these things. The story of lactate, as I said in my first slide itself, is 2.5 centuries old. Scheele in 1780 discovered actually lactic acid in sour milk. Berzelius initiated the pathology of Berzelius' work on hunted stags and estimation of lactic acid in hunted stag muscles. That started actually the pathology of lactic acid. What he has observed is hunted stocks have more lactic acid when compared to their normal counterparts. Well, several authors during 19th century, in the entire 19th century was concentrating on only lactic acid pathology, both in diabetic patients, human patients, and also muscle contraction. Now, two hypotheses came into that picture. Number one is lactic acid accumulates only in dead bodies, not in living bodies. There were few reports that lactic acid circulates in patients suffering under the microbial infections or under other pathological conditions, say it as a metabolic pathological condition. Raymond Herman are credited with the a concept of electric potential and muscle contraction. There are differences between the two, but the central idea is the changes that take place in chemicals during the muscle death or during the fatigue development could be contributes to what is called acid base balances, changes in the acid base balances that leads into this thing. But one of the authentic work extensively done from 1880s to 1923 is that of Fletcher and Hopkins in 1907 together published this paper, wherein which is very popular also. In that, Fletcher clearly demonstrated that oxygen supply to muscle produces carbon dioxide, but when the rigor is developing, muscle death is occurring, lactic acid drops steeply down, carbon dioxide drops steeply down, whereas lactic acid will be produced exponentially and then stabilizes within few minutes after the muscle death. Well, this was altered by 1920s glycogen lactic acid cycle theory, where he has stated, Meyerhoff stated that during the contraction, muscles use glycogen and lactic acid that is produced is resynthesized back into this thing. Same concept was held by A.V. Hill also, who has worked on thermogenesis during this muscle contraction. He introduced the term oxygen debt, where he has demonstrated that post-exercise oxygen consumption increases and one-fifth of the lactic acid is oxidized to provide energy to reconvert four-fifths of the lactic acid to glycogen. Well, Lunsgaard in 1930s, he has demonstrated, in fact, Lunsgaard was also working with Meyerhoff laboratories for a short period before 1929 discovery of ATP by Lohmann, for which he won the Nobel Prize. But Lunsgaard, this is important, that's why I'm keeping this thing. He has stated, he has demonstrated very clearly that iodoacetate inhibits GAPDH. That inhibition does not produce lactic acid, that is number one, but it does not hamper the muscle contraction, was one of the finest work that was demonstrated, which becomes the central theme of this topic also, where glycogen metabolism and lactic acid metabolism are two independent entities that take place, and most probably lactic acid, because of the pandemic, the second slide I told you, is a paracrine fuel that is secreted by gut microbes in response to the nutrients. Different metabolites are secreted that leads to the pathology. Interestingly, Meyerhoff, who introduced actually lactic acid theory, altered his opinion on these things. Surprisingly, these reports are not highlighted. Meyerhoff's lactic acid theory is highlighted, but his subsequent works, particularly Meyerhoff revisiting Harden and Young, this thing, 1945, Hardy and Ziegler demonstrated that pyruvate kinase produces phosphoenol pyruvate, but not pyruvate. Meyerhoff re-examined that particular thing. Meyerhoff's positivity comes from that he responded and re-examined, either confirmed it or rejected the idea by doing other experiments. And in 1949, they amended the pyruvate kinase reaction, stating that Meyerhoff, in fact, re-examined three enzymes. Number one is aldolase, where he has thoroughly demonstrated in 1949, I believe, the 49, where that aldolase reaction is not a thermodynamically favorable reaction in the forward reaction. They use both TPI purified and also aldolase purified and examined the reactions. The thermodynamically forward reaction is not possible. Another important contribution is from that of Margaria et al. And in that, they have demonstrated that post-oxygen consumption actually is partition between the two, that is fast consumption and then slow consumption. During fast consumption, lactate remains stable in the blood. Okay, let me come to the lactic acid shuttles. That is a second important factor in this lecture. Lactic acid consumption. Now, this Brooks group in 1960 was analyzing actually the muscle metabolism, divided actually the cells into two types. One is glycolytic cells and oxidative cells. They stated that glycolytic cells produce lactic acid, oxidative cells consume the lactic acid, and they are converted to carbon dioxide, and then they contribute to the energy and ATP production. What this theory? These are the graphs that were produced by carbon labeling systems. So you will see that muscle lactate increases post lactic acid. Lactic acid disappearance has nothing to do with the glycogen, but carbon dioxide is evolved in these things. Interestingly, although for 6 decades, both Gladden and also Brooks worked on lactic acid shuttles, they are not free from glycolysis producing lactic acid. Well, in the past 2 decades, the focus was on actually GPCR orphan receptors. There are nearly 600 to 800 GPCR orphan receptors that are not characterized in terms of ligand and then their metabolic manifestations. There are 3 types of this thing. In the past 10 years, particularly from Priyadarshini group from California and Japanese group, several people also have contributed to this thing, that there are 2 kinds of GPCR. GPCR is a multi-domain containing actually the protein across this thing. The cytoplasmic contains G alpha, G beta, G gamma. And then once a ligand binds to this thing, G alpha, beta, gamma gets separated into these things. And each one will have what is called the role of short chain fatty acids. Long chain fatty acids have a different roles, was demonstrated by several authors. And Priyadarshini's 2016 article in detail discusses actually, she gives a good diagram also on actually this thing. In addition to these things, one thing often ignored, although it was recognized by Offerman groups in Germany, that hydroxycarboxylic acids have a differential role than the normal fatty acids. Hydroxycarboxylic acid, there are 3 receptors that are identified. Number 1 is HCR1, 2, 3. HCR1 actually is activated by lactic acid binding. HCR2 is activated by the keto acid beta hydroxybutyrate. And then you have what is called the long chain fatty acid, octanoic acid, which is another hydroxycarboxylic acid, which is activated by HCR3. The effect of these things is they inhibit adenylate cyclase, suppress cyclic AMP pathway, activates HIF2 signaling, and this partitions HIF1 signaling to HIF2 signaling. HIF2 is more tolerant to the oxygen that is essential for essential amino acid uptake. Now, this is the signaling function of this thing. So, lactic acid also has a metabolite role. Lactic acid enters the cells through monocarboxylate transporters was shown. Monocarboxylate transporter translocation requires a basicin, that is CD147, that is active. How it is activated is still an enigma that needs a research also. Somebody can take it up. And then this basicin translocate this MCT1. MCT1 is a monocarboxylate transporter, short chain monocarboxylate transporter. Lactic acid is specifically transported, but there are also reports that pyruvic acid is also transported by MCT1 inside. Lactic acid conversion to pyruvate in livers is not possible. Therefore, perhaps liver is using pyruvic acid for activating this thing. But after conversion of pyruvic acid and after it's actually ligand activated essential amino acid uptake, glutamine, branched chain amino acids, and other things will be entering through MPC1 or nearer to the MPC1 by other the pyruvic acid and glutamine entering into this thing activates glutamate pyruvate transaminase that produces 2-oxoglutarate. Right? This 2-oxoglutarate by reductive oxidation produces citric acid. Citric acid produced in this thing is transported outside through SLC25A, that is another important metabolite transporters across the mitochondria that needs also further research. And here, citric acid in the cytoplasm is broken down to acetyl-CoA and oxaloacetate by ATP citrate lyase, and oxaloacetate is reduced to malic acid. Malic acid is decarboxylated by cytoplasmic malic enzyme. I have not shown this here because of the space problem, but it produces carbon dioxide and pyruvic acid reduction to lactic acid is done in this. So pyruvic acid produced downstream of citrate is the source of lactic acid, not the glycolytic pyruvic acid. And lactic acid is exported outside. Within the tissue, it becomes a paracrine fluid between the cells of that particular tissue, but it is ultimately exported outside into the blood, which is enters, circulates, oxidized either in the same tissues or in different tissues. A separate matter. Now, two mitochondrial pyruvate carriers are recorded. Now, according in my first slide on this thing, I told you that the confusion arises between the pyruvate carboxylation and phosphoenol pyruvate reshuttling. If you re-examine 1939 and 1940 Chava's papers also, which actually this man, this ATPase and Mitchell has quoted to support his this theory, support theory that it is hydrogen transfers across the membrane, create membrane potentials, and these membrane potentials are activated only because of actually this thing. That is actually phosphate transfer is a different phenomena from membrane potential is the summary of this thing. Now coming to this particular point, you will see that this particular step is important, and insulin resistance glutamine metabolism will not be proper. There are several references I'll give at the end of the lecture that will actually hamper this thing, but this particular cycle of succinate oxidation takes place because of the branched chain amino acid, this thing. We also have reports that branched chain amino acids also contribute to insulin resistance. Now, this phosphoenol pyruvate activates phosphoglycerate pathway, and then I'll discuss more in on the Harden and Young equation in the next step. Well, I told you in 1935 Warburg described that NADP is one of the thermostable cofactor. He has also discovered that NADP oxidizes glucose 6-phosphate in a parallel pathway to glycolysis, and that produces 6-phosphogluconate. And there he ended. Because the focus and hype in 1930s was more on glycolysis and glycogen lactic acid theory, the second discovery, that is in 1938, NAD got uppermost. There he has stated that phosphoglycerate kinase that produces actually the pyruvate. He did not discuss this particular steps which were discovered only in 1945 by Meyerhoff. I am I'm giving this particular side because my concentration is on this thing. There is a reshuffling. Aldolase, I already told you, is a controversial enzyme. Is it actually reversible, or this thing, or is a question mark? But actually, what happens is by reshuffling of these, it is Lipman who demonstrated that 6-phosphogluconate is decarboxylated to ribulose phosphate. Ribulose 5-phosphate by epimerization produces xylulose 5-phosphate. Isomerization produces this thing. Reshuffling of transfer of two carbon atoms, one containing ketone groups from xylulose by transketolase produces pseudoheptulase and glyceraldehyde 3-phosphate was this thing, and these are recycled without this thing. But transketolase is one of the principal enzymes which actually initiates the reshuffling of this thing. It requires thiamine pyrophosphate. All ketone transfer groups require thiamine pyrophosphate is known to the biochemists who understand this thing. Sorry, let me go to this stoichiometry. If you look into this thing, if this recycling is doing glycerol 3-phosphate and 2,1, what is happening to this thing? There is a reshuffling, no doubt, but ultimate the stoichiometry is 3:2. What is this thing? And you have actually phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate is the product of ribose 5-phosphate and ATP. ATP transfers two of its terminal phosphate groups to phosphoribosyl pyrophosphate synthesis. Anybody who is working on nucleic acid knows that phosphoribosyl pyrophosphate is the central hub, the basic unit on which actually pyrimidine and purine synthesis will be taking place, and UTP is one of the important molecule which is produced by pyrimidine synthesis, which is also required for gluconeogenic and lipogenic programs in these things. Okay, you will see that the fructose 6-phosphate conversion to fructose 1,6 bisphosphate is a tortuous path. It requires HIF1 activation, which produces a bifunctional enzyme kinase cum phosphatase. Fructose 2,6 bisphosphate activates allosterically PFK1. Without this fructose 6-phosphate is not. Tigar is that is mutant form of this thing. Yesterday somebody was talking about that thing. It inhibits actually this thing, and thereby it shuts off this thing. Another important observation is although phosphoglucose isomerase is considered to be actually this thing, Stinkon et al. Enormously discovered actually several mutated enzymes that contribute to several pathologies. You can examine that particular paper. It is a comprehensive paper. And in this particular case, phosphoglucose isomerase will not be functional in a reverse flux. There is one question for wet lab people who want to take up this work is, fructose 6-phosphate is also produced by phosphofructokinase. Are they working independent of the glycogen hydrolysis? A question mark which we need to examine. There are some reports, but they did not examine this particular aspect. I come to this thing. We now know that, inhibition of RNA or DNA synthesis promotes gluconeogenesis and lipogenesis. They are the differentiation factors. Glucose is a differentiating factor, and that is why glucose in the microenvironment causes actually differentiation of several cytokine producing this thing. You don't require a big lab. Take a diabetic blood, put into a cylinder, into a test tube containing glucose, you will see many of the monocytes transforming themselves into macrophages. Likewise, actually, that is why yesterday when cell therapies were coming into the picture, I asked whether actually there can be a programming on actually differentiating cytokine producing and immune cytokine producing in cell therapies. That is important who want to take up these cell therapies in this thing. I am in the evening of my life. Therefore, for the future generation, I am trying to raise these particular questions. Well, this is the Harden and Young equation, which he has proposed it in 1906, where he has stated that two molecules of sugar will be converted to two carbon dioxide and two ethanol molecule, but one hexose diphosphate, fructose 1,6 bisphosphate. He called it as hexose diphosphate. It is also called Harden ester now. Stability of this thing is maintained by the phosphate availability in the cells. If phosphate depletion or sugar depletion is there, that is what happens in diabetes, alcohol production will not take place, was his conclusion. Now, glyceraldehyde 3-phosphate, in 1945, Meyerhoff revisited this reaction. This particular paper surprisingly does not appear in any of the debates on carbohydrate metabolism nor on cancer metabolism. The reason for this is it antagonizes actually the Warburg effect. Glyceraldehyde 3-phosphate is oxidized by NAD and 1,3-bisphosphoglyceric acid, and that is reduced to NADH. And Warburg's hypothesis says that, to regenerate NADH or reoxidize NADH to NAD, lactic acid is produced by these things. In this next step, 1,3-bisphosphoglyceric acid. This PGK1 is an interesting enzyme. It is found only in carcinogenic cells and in cytoplasm of actually biosynthetic cells, and it is found exclusively in actually testis. It is where actually propagation of actually cell propagation and differentiation of actually sperm cells take place. It's a continuous proliferative tissue also. Whether this happens actually in metastatic cancer producing polycystic syndromes in women is another question which wet lab people can take up to. Okay, this ATP, but as of now, we only PGK1 and PGK2 identified in somatic cells and also in testis. Well, in 1945, Meyerhoff's contribution comes in this particular case even to the problem of actually angiogenesis and lack of angiogenesis in diabetic. He has demonstrated that there is an enzyme which is actually existing in the yeast, but during the isolation, even in the cold isolation, that enzyme is lost. And that is what is called apyrase. He named it as an apyrase. Today we know that the apyrase is nothing but ATP hydrolase, which is attached to the extracellular membrane, and this enzyme plays an important role in cell survival pathways. Carcinogenesis depends upon cell survival pathways, and this cell survival pathway also actually promotes immune suppression, inflammatory activation, and then it controls thrombosis. It actually proliferation promoter, that is cell differentiations also will be taking place where these things will be taking place. This particular enzyme should be a target for cancer, and acceleration of this particular enzyme should be a target for treatment of diabetes. That is for wet labs to consider. Now, another important aberration that was introduced by Teixeira cycle is cholesterol biosynthesis takes place only. Second important aberration that was introduced by Krebs cycle is that mitochondria are functional only when they are globular. That is one of the misconceptions that takes place despite 100. We have, remember, Benda coined the term mitochondria because of the filamentous structures which he has observed under the microscope, and we have actually Parkin activating the mitochondrial fission proteins, and that keeps actually mitochondria in filamentous state. You require even for cardiolipin biosynthesis that filamentous structure. I will come to that in the next step, but let me concentrate on actually nucleotide flux requires thousands of molecules of nucleotides and turnover is high. So therefore, you require extra glyceryl D 3-phosphate that is coming. Where from this is coming? I told you short chain fatty acids which are hydrolyzed in autophagy automatically activate the phospholipases. PLA2 is and PLC is one of the PLC is one of the two of the important phospholipases identified. PLA2, cytosolic PLA2 actually hydrolyzes membrane lipids. It produces glycerol 3-phosphate and free fatty acids. Free fatty acids act as second messengers and activate inositol phosphate, protein kinase C, calcium, calmodulins, diacylglycerols, etc. All of them are second messengers. Glycerol 3-phosphate is shuttled between dihydroxyacetone phosphate and glycerol 3-phosphate and glycerol 3. So this is a triad which is catalyzed by GPD1 and TP1, and this GPD1 and TP1 molecules, they are regulated clearly by dihydroxyacetone phosphate. Accumulation of dihydroxyacetone phosphate was shown recently by 2019, I believe, in Sabatini's lab, where mTORC1 actually is activated in response to the dihydroxyacetone phosphate. And P21 is also activated in response to the cell cycle activation, and that leads to actually the activation of this particular step. This is the central step which is required for ATP synthesis and also nucleotide synthesis that decides whether proliferation should take place or not. ROS inhibits pyruvate kinase is known. Tyrosine kinases inhibit. Nitrosylation of pyruvate kinase actually converts actually pyruvate kinase into dimeric form is known. This ROS suppresses mTORC1, activates FOXO signaling. FOXO signaling activates mTORC2 assembly and activates AKT. FOXO is a transcription factor for Richter, was shown long back by Gottini et al. By Sabatini group, I believe. And you will see that this is the central carbon metabolic pathway. Initially, it was called as one carbon metabolic pathway because glycine was contributing methyl group for the production of thymine and then DNA. That is how actually P53 got its name as the guardian of the genome. In this case, you will see that, this is the central enzyme which is actually inactivated in somatic cells. But during actually mRNA synthesis, which are required in somatic cells, this is regulated. Parkin's function is regulated by cyclic AMP and pKa signaling, which we will be discussing later. That actually allows mRNA synthesis. But P53 during the cell cycle progression, it is only halfway through when cyclin D and CDK4 complex function is over, P53 is activated in cell cycle. And that P53 inhibits Parkin in cytoplasm, activates this particular pathway. This pathway is a central carbon pathway. It synthesizes phospholipids in Kennedy pathway. It contributes to methylation groups. We now know methylation and demethylation either opens up or closes chromatin materials. And also it has an important role in creatine biosynthesis, nucleotide biosynthesis, and glutathione biosynthesis. Glutathione biosynthesis and serine biosynthesis, and also creatine biosynthesis and activation of these things require cysteine and essential amino acids. This is suppressed in mesenchymal cells. Yesterday's question to that lady from Calcutta was actually what are the markers. This is one of the important marker where TGF beta 1 will be suppressing SLC3A2, that is CD for 4F2HC, right? This particular one needs glycosylation, and that glycosylation is activated by pentose phosphate pathway. This pentose phosphate pathway converts by inhibiting phosphoglycerate isomerase. It requires glutamine. In a sequence of reactions, it actually produces UDP glycase. Here you will see that UTP is required. So before actually pyrimidine synthesis takes place in this thing, when this UTP is suppressed, production is suppressed, that is phosphoribosyl pyrophosphate synthesis suppressed, gluconeogenesis or glycogenesis takes place in these things. Let me go a little fast. Well, so I told you that, Lunsgaard himself mentioned it, and pandemic clearly shows that, lactic acid conversion and glycogen hydrolysis are independent. They are not clubbed together. How does it take place? Let us examine glycogen hydrolysis, which I have discussed in detail itself, that the production of glyceraldehyde 3-phosphate is important, and dihydroxyacetone phosphate flux comes from what is called hydrolysis, autophagy. That particular PLA2 activation leads to this DHAP, and that leads to actually increased flux. So as long as nucleic acids are important, nucleotide synthesis is important, this pathway is not inhibited. Once lactic acid enters into this thing, that is producing phosphoenol pyruvate and inhibits this thing. One question which Harden confidently did not analyze is the source, the precursor molecule of lactic acid. That remains a confusion even in alcohol production industries, even now also. In a monograph, I read the second edition. First edition I couldn't read. In second edition, he devoted nearly 3 chapters on the precursors of alcohol production, analyzing, summarizing several of the metabolites, including folate, lactic acid, pyruvic acid, blah, blah, blah. All these things, but ultimately he converged upon his own work along with Young and Young in 2012, and stated that DHEAP alone could be the precursor of alcohol production. But he was also cautious that it requires much bigger work. Today, we know that even in rigor mortis, after the lactic disappears, muscle softens up, and there is a forensic identification today that lactic acid production starts. So lactic acid production takes place in human livers is also recently reported. 1 or 2 reports I have seen, but there is no actually this thing, but there's a confusion in alcohol production. Let me come actually how lactic acid entry comes into this picture. I told you already about the signal pathway and also lactic acid shuttle. Here you require essential amino acid tryptophan to maintain the NAD levels. Kynurenine pathway gives that thing, and that produces NADH and this thing. This pyruvic acid by a reverse flux produces actually phosphoglycic acid. Another question for the wet lab people who have funds and laboratory facilities is whether this particular reverse flux converges on 1,3-bisphosphoglycic acid or stops at the 3-phosphoglycic acid. My hunch is it stops at 3-phosphoglycic acid because that diversion to serine glycine biosynthetic pathway is important in producing actually materials required for biosynthetic these things. Unlike the present model, which considers Krebs cycle as the center of actually ATP production. ATP is produced by PGK1. The role of actually the mitochondria is to activate respiration, mitochondrial respiration, activate thermogenesis, and that thermogenesis leads to what is called heat shock proteins. Heat shock proteins are chaperones. Experiences from the HIV and also malarial parasites, and then the mycobacteria, both in leprosy and also tuberculosis. Although people are not hearing that thing is, many of the actually the secondary metabolites that are produced in these things are reincorporated into what is called cell structure functions. Capsid formation is important for viral propagation, and capsid formation, capsid synthesis requires chaperones to assemble proteins of viral proteins into the capsid for propagation of the virus. That was shown very clearly that thermogenesis is important in these things. So lactic acid producing microbes activate lung cancer, and also these things are very clearly known. Short chain fatty acids, which are the byproducts of high fat diets, and also obesogenic lipopolysaccharide binding proteins, branched chain amino acids, they are actually important in these things. These are some of the references which will give you actually interlinking of metabolites entering into the mitochondria, leaving from the mitochondria. So both pyruvate carrier proteins and SLC25, which actually export citrate and other metabolites out of the mitochondria, play an important role. Thank you for your patience. I'm sorry if I exceeded my limit. Any questions? I'm prepared to answer.
Yes, sir. It's a long presentation, sir. Yes. Any questions? Any questions to Professor Lakshmipati?
Hello.
Yes, sir. Go ahead.
Hello.
Yes, this is Dr. Sundaresan, sir. Yesterday I did a keynote on the topic, and your topic is very extensive, and we are very much thankful for your extensive lecture on the biochemical variables on.
I have some audibility
osteogenesis.
problem. Can you also type your this thing on this thing? I'm able
Okay.
to make it up, but can you type your this thing, or
Yes.
coordinator can raise that? I'm able to hear him properly.
Okay. Thank you, sir.
Okay.
Sir, you can text in chat box, sir. I can ask two question to Lakshmipati, sir.
What is the question?
So, sir is going to type in the chat box, sir. So meanwhile,
Okay.
yes.
Okay, we are stopping now.
Yes, sir. Meanwhile, we will go with alternate speaker.
Okay, they can send me questions. I'll participate. Anybody is interested in some of the wet lab questions which I have raised, I'll be helping them in actually theoretical background. Thank you very much for giving me this opportunity.
Yes. Thank you so much, sir, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. On behalf of organizing committee members of Virtual Cancer Summit 2021, we want to thank you for instant presentation. Looking forward to meet again next time.
Bhaga?
Yes. Next, we ask Dr. Joshna Singh, Cancer and Neurobiology Laboratory, Department of Biochemistry, Institute of Science from Banaras Hindu University from India. The topic will be modeling epithelial cancer in Drosophila melanogaster. Yes, madam, go ahead by sharing your slide. Yes, madam, go ahead.
I'm audible.
Yes, madam.
Myself, Josna Singh. I may I start?
Yes, madam. Go ahead.
First of all, thank you Innovine Scientific Meeting to for my participation. I myself, Josna Singh. I am pursuing my PhD from Banaras Hindu University from Biochemistry Department. I did my post graduation from Molecular and Human Genetics Department, Banaras Hindu University. Today, my poster presentation topic is modeling epithelial cancer in Drosophila melanogaster. First of cancer is a group of disease characterized by uncontrolled growth of cells. Epithelial tissue is most common origin for development of cancer worldwide. According to WHO, the most frequent diagnosed type of cancer is breast cancer for female and prostate cancer for male. Now, cancer is the abnormal and controlled growth of the human health normal cells. This is the representation where normal cell get transformed into normal cell into cancer cells. This is the classification for cancer. Basically, four types of cancer. First is carcinoma, sarcoma, lymphoma, and leukemia. Carcinoma is the epithelial origin, and most of the cancer cases are mostly 85% occurrence of cancer is due to carcinoma, like breast cancer, cervical cancer, lung cancer, and prostate cancer. This is the time scale showing the key milestone and laboratory in the history of the Drosophila cancer. After 100 years, what are the key modulator involved in a tumor growth is remain unanswerable. There are several model organisms used in a cancer like Drosophila, C. elegans, zebrafish, yeast, and mouse. But we are working on a Drosophila because it is easy to culture and RNA interference, genetic engineering is easy to done in a Drosophila, and there is no ethical issue with Drosophila. This is the life cycle for Drosophila. In total 14 days it took for transform into embryo into adult life, and I'm working on wing Delport, and this is the where this is the position where wing is emerged out from the Drosophila. Why we took Drosophila for modeling the human cancer? Because Drosophila whole genome are sequenced and many cancer associated genes are conserved between Drosophila and human. And many signaling pathway are first studied in fly. After that, several study is done in human. There are different types of cancer which are used in a Drosophila for study in cancer research, like colon cancer, brain tumor, lung cancer, etc. I'm working on a script gene, which is the tumor suppressor gene, and there are several tumor suppressor gene and oncogene which are homology with human mammalian system with a fly gene. In fly Drosophila, basically, fly tumor is classified into two categories: hyperplastic tumor and neoplastic tumor. In hyperplastic tumor, the number of cell proliferation is increased without disrupting the cell cells. But in neoplastic tumor, after generation of tumor, it complete lost the morphology of cell. As we show in this slide, we can able to see that one normal cell get transformed into a new cell which is disrupted cell morphology neoplastic, but hyperplastic the cell is not disrupted. I'm working on a script gene which is a tumor suppressor gene, and it regulates the polarity of cells. And script is function in a complex with the DLG and LGL, and it regulates the polarity of cells. After down regulation of it is going to cause cervical cancer, colon cancer, etc. This is the loss of representation in which we want to show that the loss of script gene, which is a tumor suppressor gene, leads to development of cancer, and this is the representation where script we knock down using RNA interference technology. and generate the cancer. But what does the molecular candidate involved behind the script downstream is remain unknown. And this is the way how we induce tumor in a Drosophila. First of all, in activating a tumor suppressor gene, it may be oncogene or tumor suppressor gene. We first of all knock down the expression of tumor suppressor gene or may overexpress the oncogene, like in this representation. We knock down the tumor suppressor gene using RNA interference technology, and overexpressing the oncogene like RAS-B12 leads to loss of cell adhesion, basement membrane degradation, increased mortality, and invasion leads to development of cancer in the using US Galco system for generation of tumor, in which we took
Yes, madam. Just
two flights.
we are unable to hear your voice. Just we can ask to speak some loudly. Unable to hear from.
Audible, sir?
We are
Voice,
your
voice is slightly low.
very low.
Yeah, but actually slides are self-explanatory. Thank you. It's a wonderful work.
Yes, go ahead, madam. See,
Most of the slides are self-explanatory. Can go. Okay.
we are getting very low sound from your side, madam. So we ask you to speak some loudly from your side.
Sir, yes, sir, I will try. I'm audible, okay, sir?
It's fine.
Okay. I am inducing cancer in a Drosophila using a US Galfo system, in which we took two fly lines, US Galfo line and Galfo line, both one I took male and another US line we took a female. After Galfo get bind with the upstream activator region and lead to down regulation of the script gene. And using this system, we can knock down the expression of gene or overexpress the oncogene and leads to development of cancer. Drosophila is a monolayer imaginary disc epithelial tissue, which is a versatile model to study genetic and cellular mechanism of the epithelial cancer. The similarities of cellular process and emerging evidence of functional conservation of tumor suppressor gene between Drosophila and mammal that support the study of epithelial tumor progenesis in flies. What is imaginary disc? Imaginary disc, Walter, Gehring, and Ernest Holder first exposed the biology of the imaginary disc. Imaginary disc has provided a useful platform for studying the fundamental aspect of cancer due to genetic manipulation are so easier because of the small genome size. And the structure that will give rise to external appendage in adult remain protected within the larvae. This precursor structure refers as imaginary disc, where we generate the cancer. There are different types of imaginary discs which are present in Drosophila. There are 19 discs in larvae. First is the sacrolabial disc, eye-antennal disc, labial disc, humeral disc, first leg disc, second leg disc, and third leg disc. Eighth one is wing disc, ninth is hydral disc, and 10th is genital disc. I'm working on wing disc because it is epithelial in origin. So I'm generating a tumor which is in epithelial origin. So we can study using this part of the Drosophila. Wing disc development in early stage of Drosophila. Basically, the whole embryonic stage divided into three parts: head, thorax, and abdomen. This is the embryonic division of Drosophila embryo, where T2 region in thorax leads to development of wing. Wing imaginary disc is an imaginary disc which consists 20 cells in embryonic stage. After the cell proliferation in larval stage lead to growth to 75,000 of cells and the complete development of this wing disc formation. And this wing disc structure region, first showing the notum region, second is the teligula, then hinge region, margin region, and blade region. This is the whole structure of the wing disc. This is completely compartmentalization into two parts: dorsoventral and anterior-posterior ventral. This is the representation. This is the interior, and this position is the posterior. This is the dorsal region, and this is the ventral region. And uh, there are basically three group of genes which is responsible for division of anterior, posterior, dorsal ventral in the wing disc. First is the ingrail gene, ingrail gene, ingrail expressing gene in posterior region and ingrail non-expressing gene in anterior region. Apteris gene, which is responsible for dorsal, and vestigial gene, which is for ventral. This is a complete representation where gene is expression. Some other gene which also control the growth of wing disc, ingrail gene, hedgehog gene, wingless gene. These are the several genes which is responsible for development of complete wing disc. Imaginary genes used as a tumor model. The discovery of melanocytic tumor like granule in a mutant larvae by breed and Stark first suggests that the flies could develop tumor. The biology of imaginary disc of Drosophila is similar to mammalian cells that involve in a cancer. Many genes first characterizing flies have subsequent been isolated in a human and mice. Like to notch, which is so homology with mammalian oncogene, which is the H notch. This is the representation which I earlier explained that US Air Force system relies to knock down the script gene in winged images, and this is the representation in B, which shows that the confocal projection showing GFP expression pattern in a third installed larval disc after driving with PTC GAL4 using a US GAL4 system. And in our laboratory, we are working on a different GAL4, which is specific for wing disc, like PTC GAL4, BX GAL4, and wing GAL4. This confocal projection showing PTC GAL4 expression in anterior posterior boundary, and the BX GAL4 represent in pouch region, and wing GAL4 represent notum and pouch region. This is the scheme which how we knock down the expression of the script in PTC GAL4. This is a schematic representation for knockdown of the script, specifically along anterior posterior boundary of Drosophila wing disc. In this showing that after knockdown of script RNA, which is a tumor suppressor gene in different GAL4, lead to different phenotypes. Like after driving with PTC GAL4, it will show complete pupil lethality after driving with script. Whereas when driving with wing GAL4, it also showing pupil lethality, but when driving with the ferret lethality. This is the different phenotypic expression after driving with tumor suppressor gene. This histogram represents percentage of pupil lethality of different PTC GAL4. In our laboratory, we took three tumor suppressor gene, script RNA gene, which is tumor suppressor, DLG is also its component, and LGL is also a part of this whole complex. Script, DLG, and LGL, all three genes is work in a complex in which we driving with script with PTC GAL4, it show 100% pupil lethality, and when drive with DLG, it show 100% ferret let depth, and driving with LGL, it not giving any phenotype. Further, we knock down after knockdown, we overexpress the oncogene. This figure represents the brown egg in mutant show embryonic death, but in wild type, embryo is hatches out, which is indicated by black arrow after 24 hours. And B represent the histogram of 100% lethality in mutant and 100% survivability in wild type. Loss of script in wing imaginary disc. This is the confocal projection showing A to C showing the PTC therefore drive with GFP expression specifically introduce posterior boundary of Ewing, where when drive with script RNAi, we knock down the expression of script, lead to complete loss of polarity of wing disk. This is the confocal projection showing the GFP expression. After that, we did Pallotin staining, which is for actin filament to represent the whole architecture of the arrangement of actin. In the wild type, there is complete. We can visualize the hinge region, blade region, and morphology of wild type is not changes. But after driving with cancer, it completely lost the cell polarity and lead to cancer development as shown in a figure D, E, or F. And the red increase indicate that staining for F actin, and blue represent the adaptive staining for nuclease representation. So this is the after driving with cancer. Earlier stage showing the not loss of any wing disc morphology, but after later stage develop whole tumor development and leads to loss of epithelial function, polarity disruption, GNK activation, and overgrowth. But what are the molecular candidates working behind downstream or upstream is remain to answer. From this, my summary of whole presentation is that epithelial cells are the source of about 90% of human cancer origin. In our work, we are referring a broad range of cancer types which are common in origin, like cervical cancer, skin cancer, breast cancer, prostate cancer, and colon cancer. Script abrogation alone in a large scale epithelial cell is sufficient to induce tumor growth in a wing imaginary disc. Loss of function of tumor suppressor gene is lead to uncontrolled growth or over proliferation of the cells. These are my references. Thank you.
Thank you very much for a wonderful presentation. I compliment this presentation. The central theme of cancer, it has three phases. Number one is actually transformation into epithelial to mesenchymal stage. Embryologically, this is an evolutionarily advantageous system for organogenesis. That is actually what is demonstrated, the central theme of this presentation. I thank the author for a wonderful presentation. This has been my theme also for a long time. Metastatic cancers adopted this organogenesis principle. Immunity hampers actually migration of cells from one stage to the other stage. Post-gastrula stage, organogenesis depends upon the migration of cells. That has been exploited by metastatic cancers. The authors have clearly demonstrated that thing, and a wonderful thing. And then after the differentiation of each tissue, it will not encroach into the function of other tissues is the purpose of immunity. That is immunity is post-organogenesis system. That is what happens by this particular apirase enzyme, which will be differentiating actually the red blood cells into vasculogenesis. And yesterday's my question on cell therapies, whether we will be in a position to reprogram actually cells, embryonic cells, bone marrow cells, to program for specific program in cell therapies is this particular presentation addresses that. I congratulate the author. Thank you very much.
Any questions? Any further questions to Dr. Jyotsna Singh? Just if you have any questions, you can text in chat box. We'll be asking behalf of you. Even after the completion of the conference. Yes, I think no more questions, madam. Thank you so much, madam, for participating in this event as a poster presenter. Thanks for sharing your tips with us. We want to thank you for your insight presentation. Looking forward to meet again next time. Yes, this was a presentation from Dr. Josna Singh. Next, we ask Dr. Hamad Mustafa Barekazai from Department of Community Nutrition from Gazanfar Institute of Health Sciences from Ministry of Public Health, Kabul, Afghanistan. The title of the talk will be The Association Between Dietary Inflammatory Potential and Gastric Cancers, a Case Control Study. So we ask Dr. Hamad to start the presentation by sharing your slide. So, is Dr. Ana Fastino from Portugal is ready with your PPT so that we can invite you? Hello.
Next session has started.
Yeah, no, he's going to start, sir. I think he's just now he left away. Again, he joined now.
Okay.
Hi.
If Dr. Singh is online, I'll.
Yes, madam. Dr. Anna, are you ready with your presentation so that we can invite you?
I will share my presentation.
Yes. If you are ready, you can share. Because
Yes, I'm ready. I will share right now.
yes, you can go ahead.
Are you seeing now?
Yes, madam. It's fine.
Okay, may I start?
Yes, go ahead.
Okay, now,
Yes, go ahead, madam. We can see your slide on the screen.
good morning for everyone. And thank you very much for your invitation to present this work today. This work is titled Animal Models of Disease, Their Importance in Oncology. Cancer is one of the most frequent diseases worldwide. In the year 2012, cancer was responsible for approximately 10 million of deaths around the world. Cancer develops through a process. It starts with a conversion of a normal cell in initiative 1. After this, the changing cell divides, and this step is called promotion. In the last step, progression, the neoplastic cells may migrate to distant places of the original neoplasia. The tumor microenvironment is composed of many cells, namely inflammatory cells. Cancer development is associated with several risk factors, including exposure to physical agents, lifestyle, exposure to chemical agents, to biological agents, and to the family history. What is the importance of the animal models, and how did they arise? In an anthropocentric attitude, man elaborated a zoological scale and took the first place in the evolution of the species, creating the distinction between humans and animals. What is the importance of animal models in biomedicine? They are crucial for disease understanding, namely to understand their etiology, mechanisms, progression, and to search for more effective therapeutic approaches that may improve the lifetime and quality of life of the patients. 70% of Nobel Prizes were awarded to research works with animal models, and the remaining 30% were attributed to studies that based their knowledge in studies with animals. Here we can see the Nobel Prize in Physiology and Medicine during the last years. The majority of the scientists use animal models in their research, and mammals were undoubtedly the most used animals for these studies. What is an ideal animal model? An ideal animal model should be simple, should be a low-cost model, and should be similar to man as much as possible to allow the translation of the results between animals and humans. How to select an animal model? To select the model, the researchers should take into account the key question of the study, the available species, the more advantageous species in terms of technical procedures and animals' welfare, should also consider practical factors, namely animals' availability, accommodation, manipulation, and equipment, and should also consider ethical questions and scientific considerations. There are several animal models available. I divided them in 3 groups: the wild animals, the domestic animals, and the lab animals. What are the advantages and the disadvantages of each one? Starting with wild animals. The wild animals, in these animals, we can observe the natural disease cycle. They are the natural host of the disease, and we may observe the pathogenicity in natural hosts. Disadvantages. Observation and recorded are limited. It is difficult to maintain these animals in lab conditions. There is the risk of zoonosis transmission. These animals may present an aggressive behavior, and the extinction risk may also be considered. Domestic animals, advantages. They live in a human-like environment. They allow the study of spontaneous diseases like mammary cancer in the female dog, and generally they are docile animals. Disadvantages. The maintenance cost of these animals. They are companion animals, and sometimes they are production animals, which may limit their use. The last one, but the most used, the laboratory animals. They are easy to maintain and manipulate. They have a short lifetime and numerous offspring, which allow the use of a greater number of animals, and they are also cheaper when compared with the wild or companion animals. Disadvantages. They are placed in a lab environment, and the conditions are induced. Making a search in the PubMed using the terms animal model, you can observe the large number of studies and increasing number of studies using animal models. The actual situation in the European Union, according to the last report that included 28 countries, more than 9 million of animals were used for scientific purposes, which represented a reduction of 2 million animals when compared with the previous report. The rodents, namely the mice and the rats, are the most frequently used animals as models of the disease. Why? Because their physiology and genetics are well understood, they are easy to maintain, They are easy to manipulate. They are relatively cheap when compared with wild animals and companion animals. They are mammals and present many similarities with the humans in terms of their anatomy, physiology, genetics, and biochemistry. In this way, the rodents have been used to study several conditions, namely cardiovascular diseases, Alzheimer's, obesity, cerebral palsy, and cancer. Searching in the PubMed using the terms rodent and cancer, we can observe a large number of results, more than 500,000 of studies. Considering the importance of the animals for research and also the animal welfare, Russell and Birch, a zoologist and a microbiologist, intended to humanize the use of animals as models and define the 3 HARS principles: refine, reduce, and replace. Refine the methods to reduce the pain and stress and increase animal well-being. Reduce the number of animals used as much as possible, and always as possible to replace the animals by other methods, namely in vitro assays. After this, I will share with you a work performed by our research team, in which we used an animal model to address the effects of physical exercise on mammary cancer. Mammary cancer is one of the most frequent cancers worldwide. In the year 2012, mammary cancer was responsible for the death of more than 500,000 of people worldwide. Mammary cancer is the most frequent cancer in women and also the most frequent cancer in female dogs. It is also the 3rd most frequent cancers in female cats. Mammary cancer development is associated with several risk factors, including age, gender, race, reproductive factors, estrogen exposure, body weight, genetic mutations, and lifestyle. Actually, there are several therapeutic approaches to treat breast cancer, namely chemotherapy, surgery, hormone therapy, radiotherapy, and cancer immunotherapy. Despite this, considering the adverse effects of these therapies and their ineffectiveness, it is essential to search for new therapeutic approaches that may improve the quality of life and the survival of oncological patients. Physical exercise. The effects of physical exercise have been studied in several conditions, namely diabetes, Alzheimer's, cerebral palsy, obesity, cardiovascular diseases, and cancer. Previous studies addressing the role of physical exercise on mammary cancer revealed a decrease in estrogen exposition, a better body weight, an improvement of immune system, and a decrease of inflammation. Searching in the PubMed using the terms physical exercise breast cancer, a lot of results arise, but the modalities and the duration of the exercise are very different among studies. So this study aimed to evaluate the effects of long-term moderate exercise training on mammary cancer progression and vascularization using a rat model. For this, 50 female Sprague-Dawley rats with 4 weeks of age were used. All procedures were approved by the Portuguese Ethics Committee. Animals were placed under controlled conditions of air system filtration, temperature, humidity, light-dark cycle, and they were fed ad libitum. Animals, the 50 animals were randomly assigned to 4 experimental groups, 2 groups exposed to the carcinogen agents and methylenitroside, 1 group sedentary and 1 group exercised, and 2 control groups not exposed to the carcinogen, 1 sedentary and 1 exercised. After 1 week of quarantine and 2 weeks of acclimatization to the lab conditions, animals from MNU groups received an intraperitoneal administration of the carcinogen N-methylenitroside at a dose of 50 mg per kilogram. After this, exercised animals were trained in a treadmill at a velocity of 20 meters per minute, 5 times a week, 1 hour per day for 35 weeks. At the end of the experimental protocols, the animals were sacrificed through an intraperitoneal injection of xylazine and ketamine, followed by cardiac puncture. Mammary tumors were collected and immersed in formalin for at least 24 hours for histopathological analysis. They were stained, the samples were stained with amatoxylin and eosine and with the antibodies for estrogen receptors alpha, beta, and vascular endothelial growth factor A. Blood samples collected directly from the animal's heart were also processed for biochemical analysis. As a result, the first mammary tumor was detected in the group MNU sedentary 10 weeks after the MNU administration, and the first tumor of the MNU exercised group was detected 2 weeks later, 12 weeks after MNU administration. At the end of the experimental protocol, animals from MNU exercised group developed a total of 23 masses, and animals from an MNU sedentary group developed a total of 28 masses. Here we can observe some of the masses of the mammary tumors. Each animal presented 1 or more mammary tumors with different sizes. Concerning to the body weight, as expected, the body weight of animals from control groups was higher when compared with MNU exposed groups. It is also important to note that mammary tumors from exercised group were higher when compared with those from MNU sedentary group. Concerning to the organs weight, the spleen presented a higher weight in MNU exposed groups when compared with control ones. Concerning to the biochemical profile, the C-reactive protein levels were higher in MNU groups when compared with control ones. It is also important to note that exercised groups presented a higher levels of estradiol when compared with sedentary ones. Here we can observe some of the histological patterns observed in the histological analysis, the papillary carcinoma, cribriform carcinoma, and the comedocarcinoma. Here we have the histological classification of mammary tumors. It is worth to note that a higher number of lesions was observed in MNU sedentary group, and the number of malignant lesions was higher in the MNU sedentary group when compared with MNU exercised one. Here we can observe the immunohistochemical staining for estrogen receptor alpha and estrogen receptor beta. As occurs in the normal mammary gland, the estrogen receptor beta immunoexpression was higher when compared with estrogen receptor alpha, and the estrogen receptor alpha was higher in MNU exercises group when compared with sedentary one. Concluding, we concluded that the long-term exercise training reduced inflammation, increased the latency period, decreased the number of tumors and lesions and their aggressiveness. The exercise also improved the estrogen receptors alpha immunoexpression, suggesting that these lesions were more differentiated and presented a better response to hormone therapy. I share with you some of the works published from this protocol. And I would like to thank you again and to thank for all people and institutions that contributed to this work, and share with you some of the pictures of Alentejo in Portugal. Thank you very much.
Yes. Yes. Any questions? Any questions to Dr. Anna?
Good information on animal models. I congratulate her.
Thank you very much.
Yes, sir. Any questions? Any questions to Dr. Anna? First, Yes. Thank you very much, madam, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. Also, on behalf of organizing committee members of Future Cancer 2020-21, we want to thank you for this presentation. Looking forward to meet again next time in physical event as a keynote speaker for upcoming 2022. So this was the presentation from Dr. Ana Faustino from Portugal. Next, we ask again Dr. Ahmad Muzdaba Barikazai from Department of Community Nutrition, Ghaznafar Institute of Health Sciences from Kabul, Afghanistan. The title will be The Association Between Dietary Inflammatory Potential and Gastric Cancers, a Case Control Study. So I request Dr. Ahmad to start presenting by changing the slides. Yes, sir, go ahead.
Hello, sir. I think share the screen. Yes. I hope you see the PowerPoint.
Yes, it's fine.
Hello, good afternoon, good morning, good evening to all. Thanks for having me here. I'm Mustafa Barzeh from Kabul, Afghanistan. The title is Dietary Inflammatory Potential in Relation to Gastric Cancer in Afghanistan. The content is today we talk about the introductions, the objectives, subjects, methods, results, discussion, and conclusions. As we know, the gastric cancer is one of the most common cancer in developed countries. Gastric cancers have 2.2 times more likely to be diagnosed in cancer in male than females. According to Global data in 2018, gastric cancer is the fifth most common neoplasm and the fifth most deadly cancer worldwide. And South Korea had a highest rate of gastric cancer in 2018. According to WHO estimates, there are around 90,415 new cancer cases in 2018 in Afghanistan. And gastric cancer is the second common cancer among male and the fourth common cancer among female. However, in 2020, the gastric cancers became, the incidence became very high, and in the incidence in males, the first cancer among males and the third cancer among female in 2018. Several factors may contribute to incidence of gastric cancer, including genetics, age, family history, gender, population, obesity, smoking, H. pylori, and dietary intake. The dietary intakes have a long association with the risk of gastric cancer. There are several studies to show that the high intake of unhealthy diet it increases the risk of gastric cancer. The dietary inflammatory has been developed to access the inflammatory potential of a diet. It has been shown that it's extensively being investigated in relation of chronic conditions, including metabolic syndrome, obesity, cardiovascular disease, diabetes, and cancers. Most of studies showed that the dietary inflammatory is have a long associate with the risk of gastric cancer among adult population, and the data on very limited in link of between diet and disease in Afghanistan. Therefore, we aim to determine the association between DII and also gastric cancer in Afghan adults. Our objectives are to determine the association between DII and also gastric cancer among men. The second, to determine the association between DII and also gastric cancer among women. And the third one, to determine the association between pro-inflammatory components of DII and also gastric cancer. And the last one, to determine the association between anti-inflammatory components of DII and also gastric cancers. Our methods, the study participants, as we know, this is hospital-based control study, was conducted on newly diagnosed patients, minimum 1 month elapsed since dedication in Kabul, Afghanistan in 2019. And we hypothesized that almost 50% of Afghans population were consuming inflammatory diet. And the cases were chosen by using convincing sampling method with the pathological confirmed gastric cancer during the period month that had been referred to Jamhuriyat Hospital in Kabul, Afghanistan. Our cases are aged between 20 and 75 years, and the control participants were aged between 20 and 75 years old. That relatives of patients and controls and cases were measured in terms of each positive, negative for 5 years and 6. The individual history of any type of pathological confirmed between the cancer except the gastric cancer, and this is with the history of chemotherapy and radiotherapy due to cancer were not included in this study. About the assessment of dietary intake in our studies, common dietary intakes of study participants during a year before the diagnosis of gastric cancer in cases and during before the interview in controls were examined by a pre-test well format food frequency questionnaire. That's the first time we made the FFQ for Afghanistan with our teams. We use a specific FFQ that we designed for the current study, for all study that we use in the Ministry of Public Health. That consists of food items with standard portion size that usually consumed by our Afghan pupils. We also trained interview administrating the FFQ through face-to-face interview at the presence of individuals who were involved in the preparation and cooking of foods. The construction of DII that derived from the food frequency questionnaire was used to calculate dietary inflammatory index scores for all subjects. We calculate DII score using the approach developed by Shivafa et al. Based on 29 food parameters that we include all of them in here, from energy up to vitamins. They are included the food parameters in our studies. As we know, the statistical analysis for use, we use the population cases. For gastric cancer, we use 90% for case and 164 for controls. The cancers for probably dropouts, we use more patient with gastric cancer and controls for studying. And we use this formula for getting the sample size in our study. That for analyzing our study, where we can see for data analysis in our study, the case and controls were compared in terms of several variables to related the general characteristic. To do this, we use the student t-test and chi-square were appreciated. Then the total cutoff points of TII score were divided based on control participant. This was done to reduce error due to the possibility of changing dietary intakes in patients with gastric cancer. All participants was categorized based on these cutoff points. Comparing of general characteristics across total of DII was done by using an one-way ANOVA for continuous variable and chi-square for categories variable, and age and gender adjust for food intakes and nutrient across total of DII. We compute the use the linear models and compare it using the ANCOVA. Also, we use the binary logistic regressions was applied to examine the association between DII and gastric cancer in different models. We have three different models. First, we control for age and sex. Further, we adjust for physical activity, for family history of cancer, social economic distance, consumption of kebab foods, smoking, drug use, and H. pylori. We also adjust for body mass index in the last model to identify the obesity independence association in our stats. We found that in results, the mean age of study participant was 54 years of them, that 73% were females. To compare it to controls, and cases had lower mean of weight and BMI. And we also, for compared to controls, cases were more likely to be residents, history of H. pylori, middle-income families, low-income families, medicine use, pharma and worker, smoking, who smoke 10 or more than 10 cigarettes a day, family history of cancer, Hazara ethnicity, drug use, also physical activity, the Turkmen ethnicity and Uzbek ethnicity were more likely than controls, and also married Tajik ethnicity, Pashtun, high-income families, postgraduate education, history of diabetes, supplements, the cases were more likely than females. About the DII, higher DII was associated with the residents who with those persons that they are live in rural. And dietary habits of our study participants compared to controls, kids more likely to drinking healthy fried food consumptions, kebab foods, consume soy oil, intrameal water drinking, low fat foods, outdoor food usage, high salt intake, high fat food, intrameal drinking of other beverage, and very high salt intake was more likely than controls. And those cases were least likely. Their boiled food usage, medium salt intake, consume liquid oil, low salt intake, moderate fats, and microbe use. We also, for our dietary habit of study participant, the higher DII is associated with violet food usage in dietary habits of our study. Also, for those controls that had our kids had higher intakes, who are green and black tea, refined greens, whole grains, fish and poultry, red and processed meat, and paper had higher intakes than controls. And also our kids had consumed lower amounts like vegetables, legumes, nuts, dairy products, fruits than our controls. About the consumption of food group among study participants, those the higher DIY associated with lower intake, there are whole grains, vegetables, legumes, nuts, paper, and those that higher dietary inflammatory index was associated with higher intake or just only the fruits. And about those that our kids had high intake, like beta carotene, carbohydrate, proteins, niacin, iron, mufa, thiamine, about the comparing nutrient intake than controls. And also in comparing nutrient intakes among our study participants, those the cases had a lower intake, like saturated fatty acid, QFA, dietary fibers, zinc, vitamins, E, vitamin B12, vitamin B6, vitamin D, riboflavin, and selenium had the lower intake than our controls. And those that lower intakes of vitamin A, folate, magnesium, cholesterol, vitamin C, and total fat than our controls in our studies. About the comparing nutrient and intakes and dietary inflammatory index, the higher DII was associated with lower intakes of beta carotene, vitamin A, magnesium, carbohydrate, folate, vitamin C, and also it's started with related with the lower intakes of total fats, pure niacin, dietary fibers, saturated fatty acid, MUFA, as well as zinc, thiamine, vitamin E, B12, vitamin B6, riboflavin, and selenium. And the higher DII was associated with higher intakes of vitamin D in our comparing nutrient intakes. About the multiple adjust odds ratio for gastric cancer trial of we use after adjust for age 6 in individual, the highest tertile of our DII was were 2.75 times more likely to have gastric compared those with the lowest tertile, and for adjust the potential confounders that did not alter the association match, such as we can say possibly with the GRACE DII had approximately 3.59 times increased odds of gastric cancer than those with the lowest adherence. And for adjustment for body mass index, make the association extreme 3.75 times more likely to have gastric cancer that we are showing in 3 models for dietary inflammatory related to gastric cancer. About, as we know, the discussion parts of our studies that is very related to our study, as we know that our current studies is just the first study we want significant, as we won't see the significant association between dietary inflammatory index and gastric of cancer. We can find this association in adult population in Afghanistan. These findings support the hypothesis that people consume more inflammatory diet that have high risk for gastric cancers. That we are told before about the variables that related with the gastric and related with the related with the gastric cancer in adult population in Afghanistan. To base of our knowledge, this is the first study to examine this association in our adult. Some studies in line with our findings, like case control study in Italy, showed that a high intake of salt, meat, and salty foods were associated with high risk of gastric cancers. Another case control study in Mexico showed that salty snacks for more than 2 times per month was associated with 80% increase the risk of gastric cancer among adult populations. And also in another case control study in Israel and Iran that showed that red meat that have higher anti-inflammatory, we can say pro-inflammatory diet and dietary products increase the risk of gastric cancers. Additionally, another case control study in Gulistan in Iran showed that red meat has positive association with the risk of gastric cancer. Also in Italy showed that those people that have high eating the green vegetable, fresh fruits and whole grains associate with their decrease the risk of gastric cancer. Therefore, we just made for people that taking green vegetable and fresh fruits for reduce their risk of gastric cancer for having genetic disease. Also, another case control study in Korea that high consumption of fresh fruits and fruits inversely associated with the risk of gastric cancers. Also, we have other studies in Italy, Korea, Tabriz, Iran, that most of case control studies that showed that persons who are taking pro-inflammatory diet have more getting more gastric cancers than those with taking anti-inflammatory diet in their food intakes. Another systemic review and meta-analysis showed that the high inflammatory potential of diet was associated with 90% increase the risk of gastric cancer among all people that are taking the inflammatory diet in their intake of foods. As there are 2 studies that they are not in line with our findings. Another case control study in Belgium showed that lean meat was not related to decrease the risk of gastric cancers among 449 cases and 500 controls. Another studies in Japan, it demonstrated that consumption of such a vegetable as Hispanic cabbage can decrease the risk of gastric cancers. This was a hospital case control study in Japan. The mechanism of dietary inflammatory index for gastric cancers, as we know, the inflammatory process can lead to development and aggressiveness of gastric cancer. One of the possible mechanisms through which inflammatory potential of diet may affect the gastric cancer in upregulation of various cytokines and chemokines and the recruitment of numerous hematopoietic population to inflammate gastric tissues. Also, cytokines can stimulate the activation of inflammatory cells like neutrophils and macrophage, which when active produce inflammatory mediation, including reactive oxygen species. This is strength of our study, as we talked before, that this is the first study to examine the association of dietary inflammatory index and gastric cancer in Afghanistan. We also enrolled new cases of gastric who had been diagnosed in only one past month. Also, analysis in this study were adjusted for weight range of environment confounder to reach an independent association between DII and also gastric cancer among Afghan adult population. And we also have several limitations as this as hospital-based case control study, recall bias, selection bias should be considered, and also our dietary habits of hospital case control may be different from those that your general populations. Although food frequency situation is the most common used approach for assessing dietary index in epidemiological studies, it cannot rule out a possibility of misclassification of study participant. Also, we have not data on some components of DII, including ignol, ginger, omega-3, omega-6 fatty acids, saffron, trans fat, turmeric, oregano, rosemary, and flavonoids, which may influence our results. In conclusion, we found a significant association between inflammatory potential of diet and risk of gastric cancer. Further studies, we just need cohort studies for with large sample size to establish these associations. And also, we publish our article in Nutrition and Cancer United Kingdom. Thank you.
Yes. Yes, any questions? Any questions to Dr. Mustafa?
Is there any studies? Are there any studies actually relating it to the age-related problems? Because most of the cancer problems occur only in post-menopausal or near menopausal stages in case of women, or aged people in case of this thing, when growth factors are declining also. Are there any actually age-related changes where actually these modifications because of the diet occur in this thing?
Did you understand the question, sir? Asked by Dr.
Oh, sir.
Yes, sir. Lakshmipati, sir. Just to repeat the question again.
Pardon?
Just to repeat the question again.
Yeah, are there any age-related changes where actually these aberrations which are associated with the specific dietary patterns are taken up? Because, you know, in most of the cancers occur in case of women near menopause or post-menopause phases, or in case of males during the aging process, particularly in prostate glands and other things. So, does the dietary patterns during that period actually growth factors are declining in people? So, is there any relationship between the diet and that particular pattern?
Yes, thank you, sir, for your question. As you know that we had made some problem in our country, and we recognize at the age of a woman. Therefore, we just made some related to age in cancer, because it's very rare patients that they are coming in a hospital in Jamhuriyat hospitals. Most of people haven't allowed them to diagnose their patient in Kabul. Most of them they are allowed to go to Pakistan. About the women's, we just take the dietary intakes. We made the FFQ for first time in our country, the food frequency questionnaire. We just had some problem about the analysis of some foods, because we haven't any biggest laboratory to analyze some foods. Therefore, we have some problem about the age, especially in women.
Yes, any questions further? Just if you have any questions, you can chart in chat box. So we will be asking behalf of you. If you have any questions, you can raise the hand also. Yes, thank you very much, sir, for participating in this event as a speaker and providing a unique perspective. Thanks for sharing your tips with us. Also on behalf of members, members of the organizer committee, members of Innovan Meetings LLC, I want to thank you for inside presentation.
Thanks for presenting.
Yes. Next, we ask Dr. Miss Neeraja P, PhD student, Regional Cancer Center from Trivandrum, India. The topic will be mini chromosomes maintenance, proteins in early lung cancer detection, a case study. Yes, madam, you can start sharing your slide and go with presentation.
Is it audible?
Yes, madam.
Oh, okay. Good
Yes.
evening all.
Yes, madam.
Myself Neeraja, and I'm a PhD student at Regional Cancer Center, Trivandrum, Kerala, and I'm registered at Manipal Academy of Higher Education. So the topic of my today's presentation is mini chromosome maintenance proteins in early lung cancer detection, a case study. So about the background of the study, lung cancer is the second most commonly occurring cancer. Hello?
Yes, madam, go ahead.
Okay. Lung cancer is the second most commonly occurring cancer, according to Globocan 2020, and 18% of the total lung cancer deaths is due to lung cancer. And lung carcinogenesis is a multi-step process which includes accumulation of successive molecular, genetic, and epigenetic abnormalities, and a series of morphological alterations, pre-neoplastic and subsequent neoplastic lesions. And lung cancer is mainly divided into two, classified into two, and they are non-small cell lung cancer and small cell lung cancer. There are three pulmonary pre-neoplastic changes happening, and they are bronchial squamous dysplasia and in situ carcinoma, atypical adenomatous hyperplasia, and diffuse idiopathic pulmonary neuroendocrine cell hyperplasia. Despite the several advances in diagnosis and treatment, the mortality rate of lung cancer remains to be awfully high because it is often diagnosed at an advanced stage. So there is an urgent clinical need to develop simple, non-invasive, non-traumatic methods for diagnosis of early malignant and pre-malignant lesions. Now, studies using cells exfoliated on sputum holds much significance as it is a non-invasive, easily repeatable technique. Similar to all malignant tumors, aberration of cell cycle regulatory proteins is emblematic to lung tumorigenesis also. So deregulation of this DNA replication results in genomic instability and contribute to the malignant transformation of cells. So moving on to DNA replication and mini chromosome maintenance proteins. These mini chromosome maintenance proteins are DNA helicases that could unwind the two strands of DNA during replication. So here is a picture of the role of MCM proteins in DNA replication. And they are helicases. They are often loaded at a late G1 phase in cell cycle, but they are inactive at that phase. And after that, during the S phase, that is the replication phase, MCM proteins become active and unwind the DNA, which gets dissociated after their function irreversibly, limiting the replication to only once per cell cycle. And MCM proteins are degraded in quiescent, senescent, or differentiated cells present only in replicating cells. And since these malignant cells are dividing, we can see a higher expression. We are assuming that MCM proteins will be highly expressed in malignant cells. So we hypothesize that if the MCM proteins can be characterized and validated in the cells of sputum samples, a novel technique can be defined for the laboratory diagnosis of lung cancer. About the objectives of my study are lung bronchoscopy, biopsy, and sputum sample collection from patients with clinical suspicion of lung cancer attending the Respiratory Medicine Department of Medical College Hospital, Trivandrum. And second objective is that to study the expression pattern of MCM proteins in lung tissue samples and compare it with the established proliferative marker MEB1. Third one, to determine the sensitivity and specificity of cell cycle regulatory proteins for diagnosis of lung cancer. And the fourth objective is to evaluate the expression pattern of MCM proteins in corresponding sputum samples. Then moving on to the materials and methods, we have taken during our study period from 2019 to 2021, 53 lung bronchoscopy biopsy samples were procured, and these samples were subjected to formalin fixed paraffin embedded tissues to H&E staining, and immunohistochemistry was done in the following markers like MCM2, MCM4, MCM5, MCM7, cell division cycle proteins like CDC6, cyclin D, cyclin E, and MEB1. Statistical analysis was carried out after this immunohistochemical study. Next, total protein and total RNA was isolated from these bronchoscopic biopsy samples, that is fresh bronchoscopic biopsy samples. Western blot was carried out for quantitative analysis of this protein, and RT-PCR was done for transcriptomic analysis. From this, we have streamlined a marker, and at the same time, parallelly, sputum samples were collected from these patients. It was processed using BD cytorich red preservative. Liquid-based cytology smears were prepared, and pap staining was done. At the same time, sputum cell block preparation was also done. H&E staining was done in that for confirming the presence of abnormal cells. Then the biomarker streamlined above was applied to these samples, that is using immunocytochemistry and immunohistochemistry. Now moving on to the results, lung tissues for immunohistochemical analysis were selected by using H&E. H&E differentiated the tissues at structural, cellular and structural levels, and a total lung tissues were characterized, and a total of 32 tumor tissues and 21 benign tissues were obtained. The baseline characteristics of the population is given in the table, and it included 40 males and 13 females. Of the 32 tumor tissues, 9 were adenocarcinoma tissue, 8 were squamous cell carcinoma, 4 were non-small cell lung carcinoma tissues, 1 was adenosquamous carcinoma, 3 were poorly differentiated carcinoma, and 3 were atypical, and 4 were small cell carcinoma tissue. This is a representative image of the H&E staining in lung tumor tissue. Now, moving on, we are looking for the presence of MCM proteins and cell cycle regulatory proteins expression in different lung tumor lesions. So here we are representing the immunohistochemical staining of MCM proteins and cell cycle regulatory proteins in adenocarcinoma tissue. And it was found clearly that MCM7 was found to be overexpressed in adenocarcinoma tissue when compared to all other markers analyzed. And intense nuclear staining was observed in this marker. And it was also compared with the established proliferative marker MIB1. Next, MCM2 and 7 showed a higher expression in squamous cell carcinoma. Of this, MCM7 was higher when compared to that of MCM2. Cyclin E was also found to be expressed in a higher level, but faint positivity in the cytoplasm was also observed for cyclin E. This was also compared with the MIB1. Then in non-small cell lung carcinoma also, MCM7 was found to be overexpressed. Then in poorly differentiated carcinoma, also this MCM7 was found to be overexpressed. This was also compared with MIB1. And CDC6 didn't show any striking staining pattern in immunohistochemistry. So from immunohistochemical analysis in different lung tumor lesions confirmed the significant higher expression of MCM7 protein. Now, the biomarker potential of this protein was analyzed by comparing the MCM7 protein expression in benign lung respiratory epithelium, which was found negative for this protein. So it was confirmed that MCM7 protein has a biomarker potential. Was done after IHC staining, and nuclear staining was a criteria for positive expression. This is the IHC quantification data for different lung lesions, and it was plotted against proteins and H score. H score was calculated as a predictive intensity and distribution with ranging from 0 to 300. So here also in adenocarcinoma, MCM7 protein was found to be overexpressed in all the majority of the samples with a high mean value. In squamous cell carcinoma also, the same pattern was obtained in non-small cell lung carcinoma, poorly differentiated carcinoma, and in atypical cells, small cell carcinoma, and adenosquamous carcinoma. Based on this H-score calculated, statistical analysis was done. Sensitivity and specificity of each of the markers were analyzed along with positive predictive value and negative predictive value with 95% confidence interval. So here MCM7 was found to have an accuracy of 92.45, followed by MCM2 with an accuracy of 90.57. An ROC curve was plotted against the true positive rate and the false positive rate, and the highest area under the curve was found to have was found for two proteins, that is MCM2 and MCM7. And MCM7 and MCM2 showed a higher area under the curve of 0.949 and 0.937 respectively, which accounts for an accuracy of 94.34% in combination of these markers. Thus, we conclude that MCM7 and MCM2 showed a increased protein level expression by immunohistochemistry. Moving on to the quantitative protein expression study in fresh tissue samples also revealed a higher expression of MCM protein in tumor tissues. Fresh tissues were taken for Western blotting analysis, and we have made technical replicates and done the blotting analysis, and we have found a dense band for MCM7 protein in squamous cell carcinoma, and a mild band was observed in MCM2 and MCM7 in tumor samples, that is squamous cell carcinoma and adenocarcinoma, and very mild or low expression was found in normal tissues. And the relative expression pattern of MCM proteins was normalized with beta-actin in different lung tissues, and we have found a double expression of MCM7 protein when compared to that of normal. Now, so in formalin fixed paraffin embedded blocks and fresh tissues, we have found a protein level higher expression of MCM7 than MCM2 protein. Next, we have to know whether this protein is found at the transcriptional level for MCM7 protein in fresh tissues. So here, from the tissues were collected in RNA later solution, and next RNA was isolated, cDNA was synthesized, and RT-PCR was done with specifically designed primers for MCM7, and it was done in 6 tissue samples. We found a higher expression at the transcriptional level, confirming that at mRNA level also MCM7 protein was found to be overexpressed in tumor tissues when compared to that of benign samples. Next, this agarose gel electrophoresis image shows that lung tumor samples were positive for MCM7 and benign samples were negative for MCM7. Next, so here lung tumor tissue significantly overexpressed MCM7 protein at transcription and translation level, which was found to be negative or less expressing in its benign counterpart. So next we have to know that, can this protein be used as a biomarker for non-invasive detection of lung cancer? For this, we have collected sputum samples for 3 consecutive days. Early morning sputum samples were collected, and we are selecting this sputum sample because it is the only non-invasive recognized technique for the diagnosis of lung cancer. And usually this sputum analysis is often of low sensitivity because to get adequate cells from this sputum is very difficult. But from our lab, we have a technique to fish out the whole cell content from the sputum samples, and this has been published earlier. Then for our study, sputum samples were selected based on the morphological evaluation and monolayer, as I've already told in materials and methods. Monolayer smear was prepared and pap staining was done. Then cell block was prepared and HND was done. So here are the representative images of pap staining and HND images in sputum samples and corresponding cell blocks. Figure A represents adenocarcinoma cells in sputum cytology, and the same sample was subjected to cell block preparation, and HND staining also confirmed the presence of adenocarcinoma cells. Next, in figure C and figure D, we are seeing non-small cell carcinoma cells in sputum cytology and sputum cell block respectively. Next, in figure E and figure F, we are seeing squamous cell carcinoma cells in sputum cytology, that is pap stained, and then magnified image is seen at the corner of the image. Then in sputum cell block also we have found the presence of squamous cell carcinoma cells. Next, small cell carcinoma cells were also done. Then in cell block and smear also we have found the presence of abnormal cells. Next, so the presence of tumor cells in smear and cell block has been confirmed. So in we are moving on to the immunocytochemical analysis of MCM7 protein in sputum samples. And since we have got this tumor cells in smears, we have done for a immunocytochemical analysis. And in this monolayer smears, cell permeability was enhanced by treating with sodium deoxycholate. And so we have found a dense nuclear expression of MCM7 in immunocytochemical analysis, which is a possible result for diagnostic use of this marker in laboratory technique. Next, here we have already confirmed that in sputum cells we can express this MCM7 protein. But for this smear application, we have, if once collected, we can only do one staining. And then for any other marker staining, these smears will not be available. So we have done for a cell block analysis and immunohistochemical for the malignant cells in the sample, and then immunohistochemical staining for malignant cells of MCM7 showed intense dense nuclear expression And therefore, we have found a higher expression of MCM7 in malignant cells in sputum also. So coming to conclusion, out of all cell cycle regulatory proteins, MCM7 protein was found to be significantly overexpressed with a biomarker potential for lung cancer detection in this population. Sputum samples also showed a higher expression of MCM7, which may be utilized as a biomarker for non-invasive diagnosis of lung cancer. Upregulation of MCM7 in tumor tissues and exfoliated cells in sputum samples warrants further functional validation to see whether this can be used as a standalone marker for early lung cancer detection. So I hereby thank Director Regional Cancer Center, Dr. K. Sujathan, my guide, and faculties and study subjects of the Respiratory Department, Medical College Trivandrum, Manipal Academy of Higher Education, CSIR for my funding, and all the lab members, and after all my team. Thank you.
Yes. Any questions to Ms. Neeraja? Any questions? Just if you have any questions, you can raise your hand. We will be asking behalf of you. Yes. Thank you so much, madam, for speaking in this meeting. Yes, and providing a unique perspective. Thanks for sharing your tips with us.
Okay,
Also,
thank you so much for giving such an opportunity for presenting my topic, and thank you all for listening to my talk.
Yes.
Good
Also,
job.
on behalf of organizing committee members of Virtual Cancer 2021, we want to thank you for instant presentation. Looking forward to meet again next time in physical event if everything goes well in 2022.
Yes, sir. Yes. Thank you.
Yes, definitely, ma'am. Thank you so much. Next, we ask Dr. Shanaz S. Sharaf, the PhD student, Regional Cancer Center, Trivandrum from India. The topic will be prognostic role of immunohistochemical markers in triple negative breast cancer. Yes, madam, you can start presenting your slide. Hello.
Hello, am I audible?
Yes, madam.
Myself, Shanaz. I am a research scholar in the RCC Trivandrum, and the title of my poster is Prognostic Role of Immunohistochemical Markers in Triple Negative Breast Cancer. Now, coming to the introduction. According to Globocan data, incidence of breast cancer accounts for 24.5% of global cancer burden and has a mortality rate of 15.5%. In India, breast cancer is the most common cancer among females, with the highest number of new cancer incidents per year, that is around 14%, and with a high incidence to mortality ratio, that is approximately 15%, according to Globocan 2018. Now, TNBC, which is an aggressive subset of breast cancer that accounts for 15 to 25% of breast cancers, has a higher prevalence in India compared with that seen in Western countries. Overall prevalence rate of TNBC in India is 31%, which is quite higher. TNBC is diagnosed more frequently in younger patients. These tumors are poorly differentiated of higher histological grade and tend to recur earlier, that is during the first 1 to 3 years. These tumors have poor five-year overall survival and has a higher frequency of metastasization to visceral organs and central nervous system. Although advances in treatment modalities has brought down breast cancer mortality rates over the past decades, actually triple negative breast cancers doesn't benefit from it. TNBCs lack targeted therapy and are still treated with systemic chemotherapeutic drugs for both low risk and high risk patients. In order to achieve personalized or precision medicine, more accurate systems indicative of molecular characteristics of TNBCs are needed. And these characteristics of TNBCs highlight the urgent need for identification of novel biomarkers for predicting prognosis and precision treatment plans. The main objectives of this study includes meta-analysis of novel molecular prognostic markers of breast cancer for assessing their pooled prognostic potential in TNBC patients, immunohistochemical analysis of the markers identified through meta-analysis, correlation of the expression patterns of these markers with clinicopathological features, treatment outcome, and survival analysis. Now coming to materials and methods. First, we did a meta-analysis. We actually explored a lot of search engines, which included PubMed, Google Scholar, Web of Science, etc., to conduct a comprehensive literature search on breast cancer prognostic markers. Mesh terms as well as general search terms were used, and initially, a total of 873 studies were found, which contain information about various biomarkers with prognostic potential between the year 2000 and 2020. After evaluating their titles, abstracts, and after applying various inclusion and exclusion criteria, finally, we arrived at 87 studies that conducted univariate analysis for HR estimation, that is hazard ratio estimation of biomarkers, which was evaluable for disease-free survival as well as overall survival. And we could select 10 potential candidate biomarkers from this meta-analysis. Now, these 10 markers with prognostic potential in TNBC selected from meta-analysis was subjected to immunohistochemistry. And it was done in archival breast cancer tumor samples, which was obtained from the pathology department of Regional Cancer Center. We had selected 300 formalin-fixed paraffin-embedded archival breast cancer tissue blocks for a period ranging from 2004 to 2010. The median age of this patient group was 53.3 years. The median follow-up time was 60 months, and all clinical parameters like TNM staging, survival information, age, clinical stage, family history, etc. were recorded for statistical studies. We had selected 300 samples, and out of which 200 was TNBC and 100 was non-TNBC. Next, tissue microarray was constructed or was made for these tissue samples, and HND staining was done for marking the tumor areas. And in the tissue microarray slides, we had performed immunohistochemical staining. The 10 candidate prognostic biomarkers for triple-negative breast cancer were subjected to immunohistochemistry. Standard cutoffs were assigned for each marker from published articles. Its scores were then calculated from the staining intensity and percentage of positive cells. And this was subjected for statistical analysis to see their relation with survival as well as their potential for assessing independent prognosis. Means how they can are they potential candidates in assessing prognosis. Now, coming to the statistical analysis, we examined the association between TNBC phenotype and clinicopathological variables and the significance of the different prognostic markers using chi-square test. The association with survival was analyzed by Kaplan-Meier plot and log-rank test. Cox proportional hazard regression model was used to evaluate independent prognostic factors. A p-value of less than 0.05 was considered significant. And from this statistics, we could get, or we arrived at two proteins, or we got two hit proteins from the statistics that were significant for survival. And we had performed molecular assays or molecular studies for these two proteins in both tissue as well as cell lines, and we performed Western blot as well as RT-PCR for quantification of gene expression. Now, coming to the results, this is the results of meta-analysis. Here, the first table is for the overall survival, and the second is for disease-free survival. These are the proteins, and these are the pooled hazard ratio we got from various publications, and this is the p-value calculated by statistics. This is for overall survival, and this is for disease-free survival. Uh, and association, and this table shows the association of the selected markers with disease-free survival. Of these, E-cadherin, survivin, P53, MTA1, HIF1, alpha, CD133, Vimentin, and cytokeratin 5/6 were significantly associated with poor disease-free survival. And a p-value of less than 0.05 was considered significant. And coming to overall survival, androgen receptor, Ki67, survivin, HIF1 alpha, Vimentin, cytokeratin 5/6, and MTA1 were correlated with poor overall survival. Now, this is the figure. This is the representative figure of positive staining of the biomarkers identified through meta-analysis, out of which we couldn't, because it was archival tissue samples, we couldn't very well get the staining of androgen receptor and Ki67. The rest of the markers, positive staining of the rest of the markers in triple negative breast cancer tumor samples is represented here. You can very well appreciate the membrane, diffuse membrane staining of CD133, the membrane staining of E-cadherin, cytoplasmic staining of 5/6, the membrane staining of Vimentin, and I think somebody.
Yes, madam. Yes, go ahead, madam. You can continue.
Yeah, the cytoplasmic as well as the nuclear staining of HIF1 alpha, the nuclear staining of MTA1, the nuclear staining of survivin, and the nuclear staining of P53. Now, this is just the representative tissue microarray slide, just for now. Now coming to this table, table number 2, shows the frequency of expression, that is out of the 200 TNBC and 100 non-TNBC tumor samples, how many tumors expressed positive as well as how many tumors expressed negative staining for these markers, and out of which HIF1 alpha and MTA1 showed the most frequency, means in almost more than 90% of the cells, MTA1 was positive, and in more than 70% of cells, HIF1 tissues, HIF1 was found overexpressed. Now, this table number 3, shows the association between clinicopathological parameters and protein expression in TNBC tissues. In this, we couldn't, in our ethnic population, in our patient population, we couldn't very much get any positive correlation between the markers and the any relevant clinicopathological parameters, but we could, however, we got an association between HIF and tumor grade. a positive correlation between HIF and higher tumor grade in our population. Next, survival analysis was done using Kaplan-Meier plot to see how these proteins, which we got from meta-analysis, how these proteins correlated with survival in our population. And this table, this figure shows the cumulative survival analysis associated with positive and negative expression of the selected markers. Here you can see that P53 shows a correlation with survival. That is, overexpression of P53 shows a correlation with poor overall survival as well as disease-free survival. Also, HIF also shows a positive correlation with poor overall survival as well as disease-free survival. Survivin again shows a positive correlation with poor overall with poor disease-free survival alone. It shows only a marginal correlation with survivin has only a marginal correlation with overall survival. Now here low expression of cytokeratin 5/6 has marginal significance in overall survival. MTA1 has both overall and disease-free survival correlation with high expression. Here, E-cadherin also has a positive correlation with disease-free survival. That is, out of all these markers, the markers that consistently had a correlation with overall survival as well as disease-free survival was HIF and MTA1. P53 has a significance, but that protein was already studied in our lab, so further we didn't go much into that protein. Next, a Cox regression analysis for these prognostic covariates for overall survival as well as disease-free survival in TNBC was done. And here, MTA1 and HIF1 again correlated with overall as well as disease-free survival. And next, we wanted to see that these data, what I have already explained, is in our ethnic population and how it correlated with the TCGA database samples. We did a TCGA analysis to see the survival data of HIF1 alpha and MTA1 and found that MTA1 correlated with overall survival. That is, the high expression of MTA1 correlated with poor survival, overall survival, according to TCGA database. Now we wanted to see more about the proteins MTA1 and HIF1. So we performed molecular studies of these proteins. For that, first of all, we had developed a primary cell line for triple negative breast cancer. The tissue obtained from our population, from a patient of our population. We used that tissue to develop primary cell line, and we examined the expression of HIF and MTA1 in primary, how it expresses in primary cell line. And that was compared with the established global triple negative breast cancer cell line MDA1-231, MCF7, which is an adenocarcinoma, breast adenocarcinoma cell line. And also it was the expression of MTA1 as well as HIF was assessed in TNBC tissue as well as its adjacent epithelium. And these are the expression pattern we got for these proteins. As you can see, HIF as well as MTA1 overexpresses in triple negative phenotype. It is evident in triple negative tissue, triple negative breast cancer tissue. It is evident in this MDA1-231 triple negative cell line, as well as the primary triple negative cell line we have developed. Next, we wanted to see the transcriptional level regulation of these proteins. we performed RT-PCR to see how these proteins, the mRNA level of these markers are in the tumor, as well as its normal benign adjacent epithelium, how it comes in the tumor as well as the benign epithelium. And here also we got that HIF1 alpha as well as MTA1 overexpresses in tumor tissue compared to their benign adjacent epithelium. Coming to the conclusion, we conclude that cytoplasmic overexpression of HIF1 alpha and nuclear overexpression of MTA1 in the primary tumor tissue of TNBC were found to have significance as markers for predicting TNBC survival and prognosis. And these observations warrant further protein modification studies to find out the biological explanation for the overexpression of these two proteins in triple-negative breast cancers of Indian women. Thank you.
Any questions to Miss Shanaz Sharaf? Any questions? Just if you have any question, you can raise your hand. We'll be asking behalf of you. So thank you very much, madam, for speaking up. I think someone has raised the hand. Sir, good data from you. You are getting compliment from.
Thank you so much, sir.
Yes. Thank you so much, madam, for speaking up this meeting and providing English perspective. Thanks for sharing your tips with us. Also, on behalf of organizing company member of Virtual Cancer Summit 2021, we want to thank you for instant presentation. Looking forward to meet again next time, madam.
Thank you so much, sir.
Yes. Next, we asked Dr. M. Constantine Samon, the Director of Research at the Michael de Grote School of Medicine, University of McMaster University of USA. The title of the talk will be Adiposity in Survivors of Childhood Brain Tumors, the Canary in the Mine, as one of the keynote speakers for the conference. Yes, sir, go ahead.
Thank you for the invite for this meeting. I want to thank the organizing committee for inviting me. And I'm just going to share my screen so I can put the talk up here. Can you see my screen?
Yes, sir.
Great. Let's push it up here. Great. So I work at McMaster Children's Hospital, which is a tertiary pediatric academic center in Ontario in Canada. And the hospital serves about 2.3 million people, which is about 8% of the Canadian population. And my program of research is focused on understanding the early determinants of obesity and diabetes risk in high-risk pediatric populations. And one of the populations I'm going to be talking to you about today are the survivors of childhood brain tumors, which is an emerging population in the world of cancer survivorship. I do not have any conflicts of interest to declare. And today I would like to describe the phenotype in pediatric brain tumor survivors in relation to type 2 diabetes, and to examine the possible determinants of diabetes in this population and examine whether there are any current therapies that can help us deal with obesity as a main driver of diabetes in this population. It is no secret that the world is going through an epidemic of type 2 diabetes. About close to 380 million people have diabetes. Most of this is type 2 diabetes. And by 2035, there's going to be close to 600 million people with diabetes, which represents about 9% of the world population. It's important also to know that type 2 diabetes is not necessarily symptomatic early, so sometimes there's a delay in diagnosis, and the number of people with type 2 diabetes is actually increasing in every country, and perhaps even the numbers we have are an underestimate of what is happening in the real world. And an important number here to look at is actually 21 million live births are being born to mothers who have diabetes, whether gestational or pre-existing diabetes. And that data is almost a decade old now. And these children are at risk of developing diabetes and obesity later on in life. So this is a huge problem that the world is facing, and all healthcare systems are struggling to try and contain it. The brain tumor population in children, it's thankfully a relatively rare diagnosis, but children do get brain tumors. And in North America alone, about 5,400 kids get a brain tumor diagnosis. But because of advances of treatment and diagnosis have actually improved their survival rates. And when we started this project several years ago, we were thinking about data related to premature mortality in this population. But the cancer survival rates are sometimes 80% in certain tumors. And the question was, why are these kids dying prematurely? So in this study, we looked at a relatively benign brain tumor, which are gliomas. And you can see in this diagram that on the x-axis, we have the years from diagnosis and completion of treatment of the tumor and the relative survival ratio. And regardless of the age, you can see a consistent downward trend of survival in these children who have cured their brain tumor, such that by about 30 years, 50% of them are deceased. And that's a very dramatic number, because we are saving lives by treating those tumors, and yet actually mortality has been pushed out as these patients reach middle age. The oldest patients in this data set that included 6,000 children over a 40-year period was around 59. So these kids are, when they get to adulthood, they are dying prematurely. And more data started to accumulate about the risk of death in this population. And one of the things that we see here in the dark blue line here is that circulatory disease, cardiovascular disease, has increased over time, and it increases over time away from survival. There is still, of course, toxicity from chemotherapy and radiotherapy that occur, hormonal deficiencies, changes in lifestyle. But this was a dramatic change in that cardiovascular disease actually is accounting for close to about a third of mortality in this population, and it's almost double their risk of death. Several years ago, again, another study came out from Scandinavia, which included close to 8,000 survivors, and demonstrated that the risk of type 2 diabetes is about twofold higher in this population than it is in controls. And this was an important impetus for the work we were thinking about, because you would think about type 2 diabetes as a disease that is driven by obesity. And it was really important for us to understand whether obesity is a factor that is playing a role here. We are doing at the moment a systematic review that is looking at the risk of diabetes in the whole population, and mostly looking at it in observational studies. And we do also see a significant rise in the risk of diabetes in this population. And we have more data that certainly links it to type 2 diabetes, but interestingly also actually type 1 diabetes has a slight increase, indicating perhaps an immune activation related to the cancer and its treatment. And of course, type 2 diabetes is about fourfold higher than it is compared to type 1 diabetes, and that actually correlates with the Scandinavian data. Now, it is known to all of us that an increase in obesity, of course, increases the risk of the deposition of the fat mass. So body mass index, the measure that we use to define obesity in adults, is in fact a good measure to assess adiposity in adults. And because obesity is a major driver of type 2 diabetes, we ask the question, do survivors have higher rates of obesity when you compare them to non-cancer controls? So we conducted a systematic review. Catherine Wang was a master's student who did this work when she worked with me several years ago. And what we see here, this is the data on non-cancer controls. And we can see that the rates of obesity in non-cancer controls in those studies that included the brain tumor survivors was close to 17%. And when we look at the data from the brain tumor survivors, again, in the blue box here, and specifically the non-craniopharyngiomas tumors, because craniopharyngiomas are tumors that are known to be associated with hypothalamic obesity, and they're known to be associated with obesity. The non-craniopharyngioma tumors, which is every other tumor, had in fact similar obesity rates to the non-cancer controls. So if you use body mass index Z scores as a measure to assess obesity in this population, you really don't see a difference compared to controls. And yet you see higher cardiovascular risk and you see higher type 2 diabetes risk. And then, so BMI itself now wasn't really explaining why these children are dying prematurely. And yet actually it is the most common clinical measure that we all use in pediatric clinics to assess obesity. So in conclusion, for the first part of my talk is that survivors of childhood brain tumors have an increased risk of type 2 diabetes. About 1 in 6 of them are obese. But obesity rates, when we use BMI measures to assert cases, were not really different compared to controls. And then the next question, of course, that needs to be answered, is BMI a reliable measure of the fat mass in children? We know that it is in adults. That's what is used regularly. And does the fat mass differ in children who survive brain tumors compared to controls? So this work was done by Helfa Al-Faridi when she was a pediatric endocrine fellow with us, did her training, and she did this paper on the body mass index Z score as a correlate of the fat mass in the body in children with type 2 diabetes. Of course, children with type 2 diabetes are rising around the world, and the increase in adiposity and obesity is a major driver. So what we see here is a comparison between a clinical measure of the fat mass called the triponderal mass index, and this is compared to the fat mass percentage measured through bioelectrical impedance. And we can see actually clinically, these two measures run together, but when you look at the body mass index Z score and compare it to the fat mass percentage, the correlation is actually quite poor. And this is an important realization that actually the body mass index Z scores are not a very reliable measure to measure the fat mass in the body in children. It's okay for adults, but it's actually not okay for children to use it. So then the question, do survivors have higher adiposity? And of course, for those of us who work in the obesity field, the adipose tissue depots in the body come in different places. The two main ones that we are familiar with are the subcutaneous adipose tissue and the visceral adipose tissue. And the subcutaneous adipose tissue is in the abdominal wall, in the buttocks area. It contains the smaller adipocytes. It's usually less inflammatory, while the visceral adipose tissue contains larger adipocytes that produce inflammatory cytokines that are associated with insulin resistance and the risk of diabetes. There are other depots in the body that we actually know very little about, things like perinephric fat, pericardial fat, bone marrow fat. Then they all are contributing probably to a degree to metabolic health, but we just don't understand their role really well because, again, some of the access issues to those tissues are really important. And adiposity and diabetes are a dyad. So in relation to different lifestyle factors or genetic factors or epigenetic factors, diet, of course, being a big one, the excess body fat accumulation results in a chronic inflammation by activating the immune system and insulin resistance. Both of these lead to metabolic compromise, including the metabolic syndrome, sleep apnea, and other factors, and that then can lead to type 2 diabetes. The relationship is not linear. There are multiple factors that intervene in this risk, and there are multiple things, including lifestyle intervention programs and pharmacotherapy, that can alter this risk. But this is kind of the path on which these children walk after the cure of their brain tumor. And several years ago, we launched the Can Decide study. This is a cohort study that's running in several Canadian centers and collecting longitudinal data. It is a 50-year study that will hopefully address some of those questions moving forward about metabolic health in survivors. We've established the feasibility of the cohort in another publication, and we concluded that we actually need more centers to be included so we can meet our inclusion criteria and recruitment criteria. And one of the pieces of work that came out from the candidiasis study, again, work done by Catherine Wang when she was a master's student in our group, was looking at the amount of fat mass in the body and assessing that and comparing it to the kids who have no cancer. So in this paper, there were children who have a variety of brain tumors. Brain tumors are quite a heterogeneous group of tumors, depending on the cell origin. But there was a variety of tumors. Many of them, in our case, were gliomas. These are considered low-grade tumors. Treatments are usually quite effective. The brain tumor location was split evenly in the two compartments: supratentorial, meaning it's in the hypothalamic region, infratentorial is below that. And the treatments were quite variable. You can see that the majority of kids got surgery, about 40% got radiation therapy, and about half of them got chemotherapy in various combinations. The treatment in this group, again, the percentages are reported here. Kids with the brain tumors get steroids on a short-term basis to deal with nausea or to deal with headaches or increased intracranial pressure, but steroids are actually not part of the usual protocol for treating those children. In those children, about a quarter of them had endocrinopathies that you can see here: hypothyroidism or growth hormone deficiency, hypogonadism, precocious puberty, some adrenal insufficiency, diabetes insipidus. All these treatments were adequately replaced for at least a year before these kids actually joined the study. So there was no indication that there's any hormonal deficiency that might explain what we're seeing. And in this busy table, I would like you to focus on the red box here, where we looked at the fat mass percentage using bioelectrical impedance. We measured the waist to height and waist to hip ratios, which are measures of central adiposity. And I would like to, again, focus the attention on these two columns here, is that the total fat mass was higher in the brain tumor group when you compare it to the control group. The visceral adiposity measure, or the central adiposity measures, also trended higher. Waist to hip ratio is a more robust measure of central adiposity compared to waist to height ratio in children. And you can see that there's a significant difference between the adiposity masses of both the cancer controls, the cancers versus the controls, and the adiposity, in fact, is higher in girls in both the groups than it is in boys. So this was one of the early indications that the fat mass is actually different between these two populations. And we looked then at the determinants of the fat mass, and we looked at variable sociodemographic factors, the treatment-related factors, lifestyle factors, and really the two things that stood out, including a brain tumor location in the supratentorial region, that's in the hypothalamic area, and we know that the hypothalamus is a center that controls appetite and satiety, and radiation therapy. And that radiation therapy, of course, is important because, again, it can impact the hypothalamic region, and it has other effects on metabolism. But these are the two main factors that were related to the change in total adiposity. Interestingly, none of them actually were related to the central adiposity. It was mostly related to the total adiposity, but not central adiposity, indicating that the reasons why visceral fat or central fat depots accumulate is probably more complex than just the treatment factors or location of the tumor. But there was no difference that we could detect here based on waist to hip and waist to height ratio, and more detailed measures, things like MRIs, of that area is necessary, perhaps, to dig deeper into this question. Catherine went on to do a systematic review to actually assess the literature, and you can see our studies included here. There were only two other studies in the past that looked at the presence of excess fat in the body, and we discovered that there's about a 4% increase in the amount of fat in the body in those children. And you can say, well, 4% is not a huge. However, if you recognize that the total amount of fat in a child is about, an average child is about 20 to 24%, this represents up to 20% more fat in the body. And so these kids are more adipose at the presence of similar BMI measures. So if you look at BMI only in this group, or BMI Z scores, you actually miss the fact that they are more adipose. And that's really an important point, because the future cardiometabolic risk can be defined by increased adiposity. Catherine also went on to look at measures of central adiposity. And as we have seen in our study and in those studies, again, there's about 6% more adipose tissue in central adipose tissue in those children compared to controls. This was verified further by another student, Danielle Sims, when she did her thesis work with our group, in that we looked at another clinical measure of the fat mass called the triponderal mass index. And we demonstrated again that these children do have increase in their fat mass, as you can see here. And they correlate very tightly with the measures from the bioelectrical impedance. And again, the triponderal mass index, which is the clinical measure of the fat mass, was actually correlated also with the waist to hip ratio and the waist to height ratio. So that gives us a good clinical measure that we're trying to use more often now to try and measure the fat mass clinically in the body. Now, one of the things, of course, that are important is the expansion of the adipose tissue itself doesn't necessarily indicate a change in function. And the question we asked whether the adipose tissue function was different, and that is this tissue more inflamed? Is it secreting different cytokines? Is it the metabolic function of adipose tissue has been altered, regardless of the size of the fat depot? And of course, in the world of obesity, adipose tissue cytokines run in a balance that it tries and mitigate the effect of inflammatory cytokines. So in the white adipose tissue that expands with obesity, immune cells infiltrate the tissue and start to secrete inflammatory cytokines. And these are pro-inflammatory cytokines that you see here on the left side are the ones that are thought to cause insulin resistance and drive inflammation further in the adipose tissue. On the other hand, whenever there's a pro-inflammatory response, there's an anti-inflammatory response that rises, and it includes the adipokines that you see on the right-hand side of the diagram. And that seesaw function of the inflammatory and anti-inflammatory cytokines is what the body tries to do in order for it to control the inflammation that happens with adiposity increase. And in these papers, again, Danielle worked on this paper that looked at leptin. Leptin is an adipokine that is correlated with the fat mass in the body. So more leptin means more fat in the body. And you can see that the relationship is quite strong between the fat mass percentage and the log leptin. And this is just the analysis of this data. And then when we looked at the adiponectin, which is negatively correlated with the fat mass. So when you have more fat, you have less adiponectin. And this work then was by Rebecca Ronsley, who was an oncology fellow at British Columbia, who did this work with us. You can see actually they negatively correlate. So it appears, and again, this is the analysis of the data, it appears that the function of this adipose tissue is similar to that of non-cancer controls, and that the biology that drives its function appears to be similar. And for that reason, again, more work is necessary to study these cytokines and chemokines to understand whether function is actually different. But from a mass perspective, this adipose tissue, the early indications are that it functions normally. The final question I want to get into really is, are there any treatments to target obesity or adiposity in this population? Remember, 1 in 6 kids are still obese based on the measurements that we have, and whether there was any treatments that are effective, that existing, that we can adopt for our patients. And in this systematic review, again, that was done with Catherine when she did her work with us, she looked at pharmacotherapy and lifestyle intervention. Lifestyle intervention was very short-lived. There's only one study that had only 39 patients that looked at changes from baseline in BMI, and that wasn't really very significant over that time. The pharmacotherapies included drugs that are responsible for a reduction in satiety and some insulin sensitization, reduction in insulin production. All of them, if you look at the sample sizes, they were really tiny. The duration of follow-up was small, and none of them were really significant for those that reported their results. And interestingly, more recent data looking at bariatric surgery, again, very small numbers, a total of 12 patients so far, including adjusted bands and then other forms of surgery. And the weight change is significant, mostly with gastric bypass. The others really didn't hold, but the gastric bypass surgery appears to be a promising treatment. However, the selection criteria for which patient benefits from this, at what age do you do something like this, considering that these kids are surviving longer, really is unclear to us. So this remains experimental therapy at the moment, and the search is on for pharmacotherapies or other intensive lifestyle interventions that can in fact help. Now, with all this evidence that we have just evaluated, I have to tell you that when we analyzed the strength of evidence, we were very unimpressed because the overall quality of evidence was very low. The risk of bias was quite high, and there were many, many issues with these studies that made us recommend no treatment from this list. And that's why we are now in the process of assessing new treatments within our group to try and address adiposity and obesity. So these are, again, the limitations. You know, there were no RCTs so far that has looked at this. There are no long-term follow-up data. There's no randomization, and there are no control groups that can help us address these questions in addition to the small sample size. Now, I wanted to touch very briefly on why these kids do become overweight and obese. And one of the issues, of course, that happen with these children, remember, these are precious children. They have survived a serious illness. So they are sometimes excluded from activities in a school for fear of we don't know what, but they are not necessarily allowed to participate fully in life at times. Sometimes with hypothalamic damage, there's a change in energy intake. Sometimes there's a change in lifestyle itself with access to more treats. There is reduced mobility, sometimes in physical activity because of a neuropathy or pain or depression or balance issues. There's reduction in basal metabolic rate with hypothalamic damage. There's hypothalamic pituitary damage with hormonal deficiencies that require more support. And the hypothalamus, of course, has satiety and hunger centers that are damaged sometimes with the treatments. Sleep problems. There's evidence that some of these children, their sleep is disturbed. Sometimes they have vision problems, mental health problems. And of course, medications as well. Things like antidepressants that are given to those kids can also make them gain weight. Our suggestions for really the treatment in the absence of this high quality evidence is to focus on intensive lifestyle interventions, try and personalize the treatment of comorbidities to those children, aggressively manage comorbidities like dysglycemia, dyslipidemia, high blood pressure, and other ones, manage hormonal deficits very aggressively. And of course, we still need good interventions to manage adiposity in survivors. So in summary, survivors do have excess adiposity with equivalent BMI profile to controls. If we use BMI to diagnose obesity, we will miss some of these patients. And the total adiposity appears to be driven by radiation treatment and a tumor location. The visceral adiposity determinants are still unknown to us yet, and there's no high quality evidence for treatments of adiposity in this population. So the questions we're asking at the moment, what are the determinants of these adiposity measures? And the canary in the mine really, can we find interventions that are safe and effective to prevent a future cardiovascular disease? Is this early adiposity the canary in the mine? Is this a predictor of a future cardiometabolic risk in those children? And if we treat it now, whether we reduce that risk in the future? That is a great unknown, and that's what we're hoping our studies will help establish over time. And this work, again, I want to thank the group that did this work. This group is not, it's not one person, it's many people doing many things over several years. And I would like to thank everybody here for their effort. And we have many collaborators, both on the bench and in the clinical world, that have helped us make this project a reality and across Canada and internationally. And I also would like to thank the funding agencies. Without them, we wouldn't be able to do this work. So thank you very much again for your attention. I'm happy to take any questions that you may have.
Yes, any questions? Any questions to Dr. Constant Saman? Just if you have any questions, you can raise your hand. We'll be asking behalf of you. Is Dr. Sir Lakshmipati sir is going to ask a question, sir? Just we got a hand raise from your side.
Pardon?
Are you going to ask any questions?
No. It's a wonderful presentation. A wonderful presentation. Thank you.
Yes. Yeah, this was the keynote presentation from Dr. Constance Samayan. Thank you very much, sir, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. Also, on behalf of organizing committee members of Virtual Cancer Summit 2021, we want to thank you for instant presentation. Looking forward to meet again next time in physical event if everything goes well in 2022.
Great. Thank you very much.
Thank you very much, sir. Have a nice day.
You too as well. Thank you.
Yes. Next, we ask Dr. Pavitra Balakrishnan from Tbilisi State Medical University from Georgia. The topic will be the epigenetic modifiers in ovarian cancer. So I ask Dr. Pavitra Balakrishnan to start the presentation by sharing your slide.
Yeah, hello. Hello everyone. I'm a final year undergraduate medical student Pavitra Balakrishnan. I'm from India, but I'm currently studying in Georgia, which is an Eastern European country. So I'm sharing my presentation. Let me know if you can see. Is it visible now?
Yes, madam. It's going to start. Yes,
yeah, sure. I'm actually here to present a short review about epigenetic modifiers in ovarian cancer. It is not a clinical study, but it's based on clinical studies which have been conducted previously. It's a short review. So first I will introduce about the epidemiology of the ovarian cancer, which is a major problem all over the world. And ovarian cancer is the leading cause of death by gynecological malignancy. It has poor prognosis, and it takes a lot of time to show any kind of symptoms. Because of that, there is a very poor 5-year survival rate. And you can see in the picture that about the various economic zones having various prognosis rate and various incidence rates. And about standard therapy for ovarian cancer, we use cisplatin with paclitaxel. And normally in this topic, I'm going to discuss about the effects of epigenetic modifiers in this ovarian cancer. So what cisplatin doing, I will just give a small introduction about cisplatin and its mechanism of action. Cisplatin is a platinum based drug. When we're administering cisplatin, it actually binds to the DNA strands and making interlinks between the interstrand links and also intrastrand links, and thereby making the strands. This actually inhibiting the various process of the cell cycle, and also it inhibits the cell proliferation also. During this process, various protein synthesis have been inhibited, and also the cell cycle has been stopped. By doing so, it actually recruits the mismatch repair protein, and the recruitment of the mismatch repair protein actually calls mismatch repair induced methylation. So when the methylation is happening in the gene, it's making more tough for the cisplatin to attach to the DNA strands and cause the damage to the DNA strand. And this drug needs a normally functioning p53 pathway, because once this cell cycle and cell proliferation is inhibited, the cell has to undergo death by apoptosis, and it needs a working p53 pathway. Otherwise, if there is a mutation in the p53 pathway, or any mutation in the mismatch repair proteins pathway, this drug can actually not work. And there have been many proofs in which there is a mutation in p53 pathway and the cisplatin resistance development. And due to this mismatch repair induced hypermethylation, there can be a suppression of tumor suppressor genes. And also, hypermethylation leaves no place for further platinum-based drug attachment, and it actually develops resistance. When we call resistance is when the platinum-based drug is not working in a patient who is having relapse of the ovarian cancer. Relapse is basically reoccurrence of cancer within 6 months of the completion of the therapy. And also the sensitivity of the platinum-based drug during tumor relapse is much reduced when we administer decitabine or any other demethylating agents, because the demethylating agents actually demethylates various tumor suppressor genes, and also it actually demethylates various ancient retroviral genes which is incorporated in our DNA. Thereby, it actually increases the immune sensitivity of the person also. And moreover, it can help hello, can you hear me? Hello?
Madam, go ahead.
Yeah, I think there have been a glitch over here. Yeah, I'll continue.
Yes, go ahead.
Yeah, so
We can hear your voice.
yeah. And moreover, patients showed more methylation changes compared to previous state that have been clinically proved by various studies. And there have been also a proof for demethylation of the proto-oncogenes or pro-oncogenes, which actually can further worsen the condition. But so far, there have been clinical studies which is proving that demethylation can actually increase the prognosis of the patients. In some of the studies, it actually showed increased overall response rate up to 40 to 74%. But the disadvantage of the study, all the studies, is this demethylating agents are working only in patients who have the relapse. It's not working for patients who have the sensitive, who have the platinum sensitivity. It doesn't much increase or decrease the overall survival rate or overall response rate of the patients when they have the sensitivity intact in them. So it increases the sensitivity where the patients have the resistance, and it is not working in sensitive patients. And moreover, heavier pretreatment with platinum drugs in a record ovarian cancer actually decreases the survival chances when we are retreating the same patient with the same platinum drugs with different dosages or different platinum based drugs itself, because of this heavy methylation changes in the DNA. Even though decitabine is a very much cytotoxic drug, it is tolerable if it is administered in a low dose. Already, this decitabine is approved by FDA to administer for this leukemia and lymphomas. But nowadays, the studies are showing it can be also administered for the ovarian cancer patients. And the dose determining studies shown that for the solid tumors, we can actually administer 50 to 90 milligram per meter squared per cycle. And this decitabine therapy especially induces the global genome hypomethylation. As I already told, it can also demethylate the proto-oncogenes. Sometimes there have been studies which is showing this is showing a worse prognosis, but it's still a ongoing debate that whether this therapy can be given with standard therapy or not. But all the studies that have shown the positive results have administered this demethylating decitabine therapy 5 days prior to the standard platinum therapy, and it has to be administered as an adjuvant chemotherapy. And also increased, and also the hypermethylation of the BRCA1 and RASS-FIA1 genes are found higher in normal tissue, even in the cancer patients. And also, if we see RASS-FIA1 promoter hypermethylation has also been observed, and there has been a significant hypermethylation of BRCA1 gene, which is almost like 60% of all ovarian cancer patients. which can also be like decreasing the prognosis, because this BRCA1 is an important tumor suppressor gene. And oncogenes like KRAS, PI3K, and tumor suppressor genes like PTEN, ARID1A are found mutated in all type of epithelial ovarian cancers. And also various immunogenic pathways have been activated during this demethylation therapy. And especially for people who have shown the progression after 6 months, they have the enriched pathways, they have the enriched cellular pathways, especially the cytokine receptor interactions, and enhanced drug transporters, and also like JAK-STAT signaling pathways. And moreover, combining this standard cisplatin or paclitaxel therapy with decitabine along with immunotherapy actually showed more response than only administering the decitabine, because as I already told, it actually increases the immune sensitivity of the cell there, by activating the various endoretroviral genes. But the negative part about this is it can induce the allergic reactions in the patients. So it has to be monitored very closely. But still, it actually increases the overall survival rate if administered in correct dosage, which is already mentioned. And apart from decitabine, we also have the histone deacetylase inhibitors. When it is combined with DNA methyltransferase inhibitors, it actually shows the better result and better prognosis. But also it has the toxicity which is doubling after the combination therapy. It also has the same side effects as the decitabine therapy. And mostly it has to be administered with immune therapy, same as the decitabine. So objectives of this epigenetic modifiers is to have the targeted and selective therapy and to have the less toxicity. And there have been various studies ongoing to find out the correct dosage. There have been so many dose defining studies, and also to reduce the toxicity, there have been so many studies conducted. And but there have also been studies which have been stopped abruptly during this projects, which actually shows Hello?
Yes, madam, go ahead.
Yeah. And we are also targeting for the less systemic effects, as we are seeing there have been a lot of generalized toxicity effect in all chemotherapeutic drugs. So it's also to reduce systemic effects and also personalized therapy regimen, and also for combination therapy. And so discussed limitation includes the toxicity, which even causes cessation of the projects, and dependency of the P53 pathway, and allergy induction. Yeah, that's it, my presentation. Thank you so much for listening. Hello?
Yes, just be online, madam.
Yeah, sure.
Yes. Any questions? Any questions to Dr.
One question here.
Yes, sir. Go ahead.
Yeah.
Number one, normally methylations are associated with the differentiation. And as I said earlier in Josna's people, the immunogenicity increase is associated with differentiation of the cells, so that, one function of one tissue is not actually interfering with the other tissue. right? In your,
Yes, sir.
I find normally two reports. When actually cancer cell, proliferative cancer cell is differentiating, they report it as it is a suppression of cancer. On the other hand, when actually
Yes,
the
sir.
mesenchymal cell EMT is activated, proliferation is increasing. Which state is your
Yes,
work
sir.
actually? Is it in a differentiation state or an initiation of proliferation state?
Sir, actually, these studies actually shows this demethylation drugs actually making this DNA more exposed to the platinum drugs. Thereby, it can the platinum drugs are prone to cause more damage to the further damage to the DNA.
Percentage of actually proliferative cell, how many of them are going down in this treatment?
That have been not shown in the studies so far. I have referred mostly this studies have been concentrated on overall survival rate of the patients and overall response rate and progression-free survival rate, sir.
Okay. You need to examine these things. There are two contradictions in this thing. I find this thing in global literature. Many of the people who where actually differentiation is occurring, they consider it as a treatment of cancer. It is not. Actually,
Okay,
it only
sir.
reduces the mesenchymal cells. Okay.
Okay, sir. I understand. I will do further study on this, sir. Thank you so much for your question and suggestion, sir.
Thank you.
Yes. Any further questions to Dr. Pavitra Balakrishnan? Any questions? Yes, thank you so much, madam, for speaking in this meeting and
Thank you
providing
so
a
much.
unique, yes, and provide a unique perspective. Thanks for sharing your tips with us. Also on behalf of organizing committee members of Visual Cancer Summit 2021, we want to thank you for excellent presentation. Looking forward to meet again next time.
Thank you so much. Thank you.
So, definitely. Within 3 to 4 working days of the conference, you will be getting officially electronic booklet as well as participation certificate through email ID, madam. Yes. Next, we ask Dr. Mohammad Jobairul Islam, Department of Medical Physics and Biomedical Engineering, Gono Vishwavidyalaya from Bangladesh. Yes, sir, you can start sharing your slide.
Very good evening, dear colleagues. This is Jubair Islam. I am medical physicist come art CEO, Lab at Cancer Hospitals and Super Specialist Center in Bangladesh, and as well as Joint Secretary, Bangladesh Medical Physics Society. Now I will talk on the design, fabrication, and validation of 3D printed specific antrum applicator for electron radiography. This is contents. Megavoltage electron beams represent an important treatment modality in modern radiography, often providing a unique options in the treatment of superficial tumors. It is offered that capacity of spare normal tissue distal to the target volume due to the rapid fall of the depth dose. And modern high-dose Linux typically provide any additional of 2 photon energy severe electron beam energy in the range from 4 MeV to 25 MeV. Electron radiotherapy is a labor-intensive treatment option that is complicated by the need for field shipping devices such as electron applicator, shielding, and cutout, etc. Electron beam applicators are usually used to collimate the beam and are attached to the treatment unit head. The application of the applicator is removing of the scattered by air when electron excited from the head of the liner. Several cones are provided usually in square field size ranging from 5 to 5 centimeters square to 25 into 25 centimeters square. However, it is difficult to use this type of field in critical patient positions such as canthus, nodes, etc. To treat this small lesion, interim applicators are already available in an area, but it is a fixed diameter size 2, 3, and 5 centimeter. However, are so expensive and are fixed in diameter, not patient specific or irregular shape. In order to solve this problem, the idea of a 3D printed interim applicator is introduced. And by efficiently using this 3D printed interim applicator, different anatomical small lesion and patient-specific lesion can be treated. So what is the 3D printing? And the 3D printing is a process making three-dimensional solid object from a digital file. This creation of a 3D printed object is achieved using additive process or additive manufacturing. In additive process, object is created by laying down in successive layer of materials until the object is created. There are different types of filaments as ABS, PLA, PS, PP, PV, PMMA as a printing material. So what are my objectives? There are three objectives actually in this work: to design and construct 3D interim electron applicator of defined shape, thickness, and density to minimize the radiation leakage, to verify and differential different material to print this applicator with a 3D printer, and to measure the absolute dose using 3D printed applicator in Linux machines. I have done this work at Department of Radiotherapy and Radiation Oncology, University Medical Center Mannheim, Heidelberg University, Germany. There is different types of materials. So the first one is the repref 3D printer, and another one is the filament wrapping Electra versus SD, and this is the existing interim applicator was the 3, 4, and 5 centimeter diameter shape, but it is the fixed shape, no irregular shape. It's available, and I'm using SolidWorks software and simplified 3D software. And this is my methodology process. At first, I design applicator using the SolidWorks software. Then it's convert to SolidWorks file to the G-code format using the simplified 3D software. And after that, quality assurance of the 3D printing it is passed. Then I have next to the next procedure. Otherwise, if it is failed, then stop. And then again, calibration this the bed and of the 3D printer. And then when it passed, printing of the application using 3D printer. Then experimental setup in Linux, control console plan imported from TPS. Then VM1 is the measurement. So what is the methods? The 3D printed process buildings a 3-dimensional objects from a computer-aided design. What is the CAD model? Usually by successively adding material layer by layer, which is why it is called also additive manufacturing. In this case, a versatile software, SolidWorks, using this 3D modeling to make this 3D modeling first sketch in a 2D shape. After this 2D shape, this 2D sketch imported and then using the defined feature and models using 3D models. And then design interim applicators on SolidWorks software. In this study, interim applicator of the length was the 10 centimeter, wide also 10.6 centimeter, and height 10 centimeter was designed in SolidWorks software. And the fabrication of 3D printed customized interim applicator. After modeling the interim applicator in SolidWorks, the model was saved into the STL format and for slicing and tool for the setup. Before I start printing, bed of the 3D printer need to be properly calibration for better treatment quality. And this is the when working, it was working in my lab. And the setup and procedure of absolute dose measurement. The 3D printed applicator was fixed in the existing applicator in the gantry, which was the 10 by 10 centimeter square, and along with the ionization chamber with making. After all connection fixing, the 3D printed tantrum applicator was fixed in the surface dose, whereas the SSD was the 95 centimeter. At first, I have completed 100% measurement without any additional slabs. Then all next slabs carried out the additional slabs from both materials with the described thickness and in the protocol. Ionization chamber is also attached with a PTW unitary dosimeter. At that control console plan important the TPS and consult executed. So what is the result? Absolute dose measurement in the gap chromic field when this is existing 6 MB using 6 MB beam. This is the 2 centimeter diameter, and this is the same as well as but it is the 3D applicator. And for 8 MeV, this is the original which is existing in the provided by vendor, and this is designed by 3D printed applicator, and the same as 10 MeV, and this is the without any applicator. But we have seen that here is we found as a ring shape in 3D printed applicator for 8 MeV and 10 MeV. And this finally result. Absolute dose measurement using. And here is the different thickness and difference energy. At first, 6 MeV energy for 2.2 centimeter. Here we have seen 0.652 gray. Where is the for 15 MeV energy 1.8 gray. And when we've seen for 4 centimeter thickness, only we found 0.01 gray in for 6 MeV. That is the too small. Sorry, this is using ABS filament, which is the actually plastic filament, and this is the metal with ABS PLA filament. This is actually metal and plastic mixed, and 1 centimeter and 2 centimeter. For 2 centimeter, we found 6 MeV energy 0.097 only for 2 centimeter. When if we increase this thickness, then we will reduce this leakage energy. And electron percent PDD. This is PDD from 6, 8, 19, 9, 12, and 15 MeV. Here is the almost 6 for 6 MeV energy. The PDD is also good, but for 15 MeV energy, the PDD is not working well. Okay, what is discussion? At first, the PDD curves measured agreed very well with calculated PDDs for low electron energy. Already I've said that, but the amount of the leakage radiation is more for the higher electron energies than lower electron energies. However, this problem can be overcome if we use the highest density filaments and increase their thickness. Absolute dose was dependent on the design, thickness, material, and infill densities. Sorry. The absolute dose was decreased when the model thickness and density increased. The 3D printing process allows the accuracy in manufactured applicator at low cost and acceptable filter fitting to complex geometry. From this study, it has been shown that it is very easy to treat using the type of the field critical patient positions such as canthus nose, etc. What is the limitation? When I worked, I've limited. It was limited because filament price is costly. I've used 2 filaments. What is the ABS and PLA filament? And another limitation was the more time required. Is for 4 centimeter tantrum applicator is it was time approximately 27 to 28 hours. When I entered the lab, is German time 6 a.m. and I leave office at 7 to 8 p.m. when it's working on third, and when I enter the lab near next day, then it will be it was finished. The future work to compare between the different types of filament and thickness to compare between the 3D printed applicator and existing vendor provided applicator. So conclusion, the studies developed a new 3D printed electron applicator and demonstrated its validity to superiority to provide accurate treatment for small lesion cancer cases using irregular and regular shaped interim applicator for lower energy and surface-based skin cancer. The method offers practical advantage in the neither patient nor staff must be present during the applicator fabrication process. Thank you. Any question?
Yes, this was the presentation from
one aspect is overall effect of acidity alkalinity changes in the tissues. Could it be examined in this? Does it vary with the thickness of the tumor?
So I'm not using this for patient purpose only. I am using it as is experimental purpose.
But ultimately, it needs to be addressed in the form of what is called
exactly.
acidity alkalinity, isn't it? If
So
that is the case in three-dimensional, in three-dimensional cancers also, is there a variability in the acidity and alkalinity? Can we localize that particular regions with specific changes in this alkalinity and acidity?
Yes. So I'm working for it, working already. My second chance to compare between the 3D printed applicator and existing vendor provided applicator, which is the 2, 3, and 5 centimeter diameter size, and using this different types of filament. When it will be then okay, then I will experiment in the patient.
Yeah, that could be a good output in case your instrument needs to be validated in that direction in solid tumors particularly.
Yeah. And it's very cost effective. When I had that is too much cost in the 2, 3, and 4 centimeter diameter type shape electron applicator. But when I using this type of electron applicator using the 3D printed, is too much cost. A small cost effective for South Asian countries as well as Bangladesh, India, Nepal, for low and for developing countries.
Thank you.
Thank you, sir.
Yes. Any further questions to Dr. Jovalud Islam? Just if you have any questions, even after the completion of the conference, we will be asking you. You can text in chat box. Yes. Thank you so much, sir, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. Also, on behalf of the members of the organizing committee, members of Innova and meetings, I want to thank you for inside presentation. Your message was exactly what you need to hear. Yes.
Okay, thank you. So, give me this floor.
Yes. Next, we ask Dr. Jetnit M. Hedinu from Division of Pharmaceutical Science, College of Pharmacy and Pharmaceutical Science, Florida, from A&M University, USA. The typing will be anti-tumors effects of flavonoids in TNBC through the regulation of RNA. Sorry. Yes, sir. Go ahead.
Can you hear me? Hello?
Yes, sir. It's fine.
Hi, everybody. Good afternoon. I'm getting it from Florida Agriculture and Mechanical University, USA. I'm a graduate assistant at the Department of Pharmacology Toxicology. And this is a published article from high impact journals, and I'm going to talk about the title and the anti-cancer effects of flavonoids through microRNA modulation in triple negative breast cancers. So this is my outline. We're going to talk about introduction, the microenvironment of the NBC or triple negative breast cancer cells, epigenetic modification and TNBC, and microRNA biogenesis and function, and type of microRNA roles in the TNBC flavonoids. And finally, we'll see the molecular mechanism of action of flavonoids. So as an introduction, breast cancer is a major and the most common malignancy type of cancer in women, and it accounts for 30% and the second cause of death globally. And it's approximately 1 million breast cancer cases are diagnosed globally, and in the US, it's almost greater than 289,000 new breast cancer cases were expected by the end of 2021. So as you can see in the chart, from when you compare the incidence of breast cancer from 1955 to 2021, it's increased from 1 to 11 to 1 to 8. Even though the mortality rate is decreased, but the morbidity rate is increased from day to day. So there are some classes or classification of breast cancer. Among this, the NBC is the one which accounts for 50% of all breast cancer cases, and it's characterized by the lack of progesterone receptor, estrogen receptor, and HER2 receptors. So this TNBC is highly common, aggressive tumor growth, aggressive clinical demonstration, and low survival rate. When we see the prevalence of this TNBC among in the racial group, African American women have high incidence of having TNBC when you compare with non-African American women. So mostly this TNBC is unresponsive to conventional treatment, and mostly they develop resistance. So we will see later how this resistance is a common challenge in the patients that have suffered from the TNBC. Also, chemoresistance is a challenge. Still, according to WHO, chemotherapy remains the primary cancer treatment approach for TNBC. MicroRNA were identified to be correlated with chemoresistance in TNBC from researches that have done before. A population of some microRNAs listed here and downregulation of some microRNAs listed here were associated with doxorubicin. This microenvironment, there is a tumor microenvironment are associated with poor prognosis of TNBC. Moving to this next one about the microenvironment of TNBC, it is a cellular environment in which a tumor cell exists, and it is the interaction between cancer and non-cancerous cells. And that's very important to control the sequence of cancer cell growth and progression. This tumor microenvironment is very essential for the cancer that determines initiation, development, proliferation, angiogenesis, invasiveness, metastasis, and tumor behavior progression. As you can see here, the tumor microenvironment that encompasses different environments that includes macrophages, eosinophils, blood vessels, RBCs, and others that are very important to help to initiate and for progression of the NBC. So this microRNA and epigenetic modification are involved in regulating various signaling pathways within the tumor microenvironment. So the epigenetic modifications, there are several epigenetic modifications that have been reported in TNBC, but among those, DNA methylation histomodification are the ones that are included under epigenetic modifications, and microRNA are the other ones that are major ones that strongly associated with the progression of TNBC. MicroRNA are important in silencing the expression of genes in TNBC, either by suppressing or activating various genes, but mostly it is very important to suppress or silence the specific target proteins. Since they are encoded in the fragile chromosome regions, microRNAs are susceptible to epigenetic modulation. That's why mostly the cancers are, there are some microRNAs that are regulated and some microRNAs that are downregulated, and altogether they are associated with the poor prognosis of this TNBC. When we look at the microRNA biogenesis and function, as you look at the graph, microRNA that are encoded by as a gene and there are processed by RNA polymerase III that forms the primary microRNA. And when processed by Rocha, they form the precursor microRNA, which contains around 60 to 80 nucleotides, and they moved to the cytoplasm using the X14 as used as an export, and they form a complex with the dicer, and finally they form a complex with the RNA-induced silencing complexes that are very important to bind with the target messenger RNA of target proteins, and finally they enable to silence the genes. So this microRNA controls various physiological functions, including cell growth, proliferation, differentiation, development, reproduction, apoptosis, and metabolism. And it is abnormal expressed in various cancers, including TNBC, which is shown in different research that I've done before. When you look at the type of microRNA and roles in TNBC, microRNA can serve as either a tumor suppressor or oncogene if there is any upregulation of this either of them, either tumor suppressor dysregulation or oncogene dysregulation that are associated with TNBC. Up and down regulation of the specific microRNAs may result in the repression of tumor repressor and gene increase oncogene expression. Oncogene microRNA, when we look at examples, mostly microRNA 155 is involved in the tumor cell growth, invasion, survival, and migration, and also epithelial to mesenchymal transition and immune response. Is the most frequent upregulated type of microRNA in breast cancer. Overexpression is significantly inhibited the tumor suppressor gene, suppressor of cytokine signaling, and leads to constructive stage 3. And overexpressed in TNBC and involved in metastasis and prognosis. And the second one is tumor suppressor. Common one is microRNA 205, which is very important to prevent tumors development by negatively regulating gene and or oncogenes that regulate initiation, differentiation, cancer cell progression, or apoptosis. And mostly it is downregulated in triple-negative breast cancer cell lines. When it is upregulation, it suppresses invasion and metastasis by inhibiting the expression of RB3 and VEGF-A. Overexpression can inhibit cell invasion, differentiation, proliferation through targeting the chemobox of D10, which enables to induce the level of p53 and suppress the level of SNL1 in TNBC cell lines. This is some selected microRNA which are upregulated and downregulated in TNBC when there is a downregulation of selected tumor suppressor microRNA, as you can see here, and when there is upregulation of some microRNAs, as you can see here, there is a metastasis, migration, radio cell maintenance, epigenetic alteration, poor survival rate, poor prognosis, and promotion of epithelial to mesenchymal transition in TNBC. The TNBC microRNA therapeutic targets, when you can see there is a microRNA or treatment of TNBC, since there is no conventional chemotherapy, and rapidly develop resistance to chemotherapy and limited immunotherapy responses. So microRNA can be used as anti-cancer drug by either increasing or decreasing microRNA levels or improving conventional chemotherapy efficacy in triple-negative breast cancer cells. Oligonucleotide analog and antagonists signify the two most microRNA therapies, but there are also other microRNAs involved in increasing tumor cells sensitivity to chemotherapy. So as we can see, microRNA in triple-negative breast cancer, there is oncomere, and there is also tumor suppressive side, but mostly it's downregulated in most TNBC. So when you look at the treatment strategy, we can downregulate the upregulation of oncomere, or we can upregulate the tumor suppressor microRNA, which is originally downregulated in TNBC. So there are drugs that mimics the microRNA, which can call as microRNA replacement therapy, or you can antagonize the upregulated oncomere, which can call as antagon microRNA or microRNA sponges or microRNA locked nucleic acid oligos. Mostly, those three are the very important targets for the treatment of oncomere, which is upregulated in TNBC. But this antagon microRNA is the one that are mostly in clinical trials, but these microRNA sponges are computed. The last one, microRNA lock nucleic acid oligos, that are very important to lock the ribose ring of the RNA or the single-stranded RNA or the single double-strand DNA, which inhibits competitively by locked the ribose ring of the base, which are finally leads to destabilize. So the third one is to increase the sensitivity to anti-cancer agents. There are several clinical studies on clinical trials that are combined with current conventional therapy with microRNA. Most studies have shown that this microRNA, which is specifically used for TNBC, that increase the sensitivity for cancers to anti-cancer drugs. So among this, flavonoids are the one that targets microRNA that regulates or the modulation of this microRNA as a potential therapeutic options in TNBC. So we look on the next slide is how flavonoids targets microRNA and used as a potential target site for the treatment of TNBC. Flavonoids, it's a natural derived product, play a significant role in the anti-cancer drug discovery field. For example, paclitaxel. So as you know, greater than 60% of the current available anti-cancer drugs are obtained from these natural products. So these flavonoids were really found in different medicinal, nutraceuticals, pharmaceutical applications, and it found ubiquitously in vegetables, seeds, fruits, cereals, and tea. Have anti-inflammatory, antioxidant, anti-carcinogenic, and anti-mutagenic properties. So their wide distribution, low toxicity, guarantees the safety for human beings. That's why most people, they use this one as prevention of different diseases, including cancer. Significant impact on cancer processes such as growth, proliferation, differentiation, inflammation, angiogenesis, invasion, and metastasis. So when we look at the class and the classification of flavonoids, mostly there is, as you can see here, this is a basic structures, but there are adjuvants that are able to classify flavonoids into 6 parts. So first one is flavonoids, the second one is flavonoids rheols, flavonols, isoflavones, flavonones, and anthocyanins. Most all these are the common dietary sources, including lettuce, grapes, teas, cabbage, tomato, wine, berries, onion, and others as a common source for these flavonoids. When we see the molecular mechanisms of action of flavonoids in the prevention and therapy of TNBC, the first one is able to regulate mitochondria. Mitochondria, as you all know, source of ATP, and also they are included to generate or balance reactive oxygen species and nitric oxide species. So this mostly in TNBC, mitochondria is impaired, and flavonoids, for example, luteolin, enhance the tumor suppressor P53 and increase apoptosis by upregulation of the specific microRNA 34-5pp and downregulation of microRNA 21. This is mostly upregulated in TNBC, but this flavonoids is very important to downregulated the upregulated microRNA in TNBC, indicating that the repression of the P53 through regulating microRNA, specifically 34A5P21, could reverse the impaired mitochondrial respiratory chain activity, which is very important, which is downregulated or impaired in TNBC. When we use this flavonoids, it is very important to reverse to normal function in mitochondria. The second one is induction of apoptosis. As you know, apoptosis is a major mechanism of action of most anti-cancer drugs, but mostly in cancer by nature, they decrease apoptosis but increase anti-apoptotic genes. So compounds which induce apoptosis represent a significant part in the treatment and prevention of tumor. many tumor suppressor microRNA are involved in the induction of apoptosis in TNBC through regulating various endogenous signaling molecules associated with apoptosis. This impact was exhibited by several flavonoids. For example, fisetin, a class of flavones, including apoptosis in TNBC cells, through activation of initiator caspase 8 and 9 and polyadiposaccharide P ribose, which is very important to regulate and induce apoptosis, but inhibiting the anti-apoptotic genes. So this flavonoid is very important as an induction of apoptosis, set as a target in the treatment and prevention of TNBC. The second one is induction of cell cycle arrest. Continuing cell cycle process is a main feature or the hallmark of cancers, and several mechanisms as controlling the process of the cell cycle to ensure proper cell division. Through the regulation of microRNA, flavonoids have been shown to enhance the cell cycle arrest in many cancers, including TNBC. These cells showed fewer cells in the G1 phase, while increased percentage of the G2M phase, suggesting that induction of G2M phase arrest, which is finally used as an induction of TNBC. The fourth one is used as an induction for autophagy. Autophagy by itself, it's a double-edged sword, have two double functions. Mostly in the early stage, it is used as an anti-cancers. And when it is in late stage, the cancer cells uses it as a mechanism to defend itself from a normal process, and also the associated with chemoresistance in the late stages. So this autophagy is a lysosomal degradation process found at low levels under normal physiological conditions to maintain cellular homeostasis by balancing catabolic and biosynthetic process. MicroRNA, including miR7 and miR409-3P, regulate autophagy through the modulation of PI3K or phosphatidylinoacetyl 3 kinase, AKT or mTOR, or mechanistic target of rapamycin. So when we see some selected flavonoids such as etigenin induce autophagy in the early stage and apoptosis and inhibit cell proliferation in TNBC cells by inhibiting the PI3K AKT mTOR pathway. The fifth one is inhibition of cell migration, metastasis, invasion, and epithelial to mesenchymal transition. So this AMT enables the tumor cells to detach from the original cancer sites and travel into the distant normal tissue, blood, lymphatic system to form metastatic lesions. Some microRNA, including late-7G, microRNA 21, miR101, miR125, F5P, miR424, miR579, and others are highly involved in tumor cell migration, metastasis, invasion, and AMT in TNBC. In TNBC, miR655 is highly downregulated, and its overexpression is strongly reduced AMT. So, fisetin is an example of flavonoids exhibited suppression of these cancer cells, growth, invasion, migration, and promoter cell cycle arrest, induce apoptosis, significantly reduce cell proliferation, and reverse AMT process by regulating the selected microRNA mentioned above. The other one is inhibition of proliferation. As you can see here, cancer cell is an uncontrolled proliferation. The body cannot control the proliferation of the cancers. So any dysregulation of this proliferation appears to be the main hallmark of susceptibility to tumors. Potent oncogenes like miR21 and miR191 are associated with increased tumor cell proliferation in TNBC. Flavonoids have been demonstrated to suppress proliferation in several human cancers, including TNBC. Curcumin is another example of flavonoids, which is a upregulation of the tumor suppressor miR34A, ultimately suppress TNBC cell proliferation. So this flavonoid is very important, as we have seen before, generally downregulated in cancer cells. But this curcumin is very important to upregulate the downregulated tumor suppressor miR734, which is able to downregulate the cell proliferation. The last one is that enable to modulate the signaling of transduction. So the conversion of external stimuli to intracellular signals mediates gene transcription, which activates cellular machinery appropriately and has a pivotal role in the normal cellular signaling process. Some of the mechanisms of flavonoids used in the prevention and treatment of TNBC through modulation of signaling transduction are the protein kinase C or PKC, mitogen activating protein kinase MAT-ERK, phosphatidylate inositol 3 kinase PI3KT, or Wnt signaling beta-catenin, JAK-STAT3, NRF1, NF-kappa and SMART3. So we will see the next slide. From this, we cannot see each of them in this presentation, but we will see some of the selected ones that are very important and crucial in this presentation. So the elucidation of these cellular and molecular events affected by flavonoids reveal novel strategies and molecular targets for developing similar acting compounds used for the prevention and treatment of TNBC. The first one is PKC pathway or protein kinase C pathway. As we all know that this protein kinase pathway is very important and is a well-known integral part of the cell signaling machinery enzyme. But there is an imbalance or impact of this pathway during TNBC or other cancer cells. These protein kinase theta and alpha isoforms are mainly upregulated growth and proliferation. However, all of these effects are reversed by PKC inhibitors. The microRNA 200B function as a tumor suppressor to reduce cell migration and invasion by targeting the protein kinase C alpha type in TNBC cell lines. The quercetin has a type of or class of flavonoids which has an anti-proliferative activity through inhibiting the signal transduction target such as protein kinase C. The second one is NRF2 KEAP1 axis. This NRF2 is a nuclear factor 2 erythropoietin factor, and the KEAP1 is the culture-like ECH associated protein 1 axis, which is, as you can see in the figure, which is found in the cytoplasm, which is bound with the KEAP1, and finally they're degraded by the proteasome enzymes. But if there is any oxidative stress, including cancer, this NRF2 is detached from the KEAP1 and translocated into the nucleus and bind with the antioxidant response element to the target gene, and finally able to express the detoxifying proteins and antioxidant enzymes, which enable us to protect the body from oxidative stress-related disorders, including TNBC. So this NRF2 provides cytoprotective activities repressing oxidative stress response levels in many cancer cells. Flavonoids such as epigallocatechins 3-gallate upregulate tumor suppressor MIR29B15P could repress the expression of NRF2, and also another flavonoid thymoquinone increase activation of NRF2, indicating that a cytoprotective effect against several diseases, including TNBC, by regulating NRF2 KEAP1 axis pathway. The third one is the PI3 or phosphatidylate inositol 3 kinase AKT emitter signaling pathway. This PI3K AKT represses apoptosis and then promotes cell survival by enhancing anti-apoptotic protein expressions and reducing the pro-apoptotic protein activity. So the PI3K AKT emitter pathway is positively associated with cell proliferation and survival, and any dysregulation of this signaling pathway significantly mediated the progression of the various cancers, including TNBC. Emitter involves induction of cell survival, cell cycle progression, and proliferation, angiogenesis, and migration. The type of flavonoids, such as quercetin, act as an AKT inhibitor that suppress the tumor cell survival, migration, and metastasis, improve the efficacy of chemotherapy, and increase the accumulation of drugs in the tumor cells, which is very important to toxic to these tumor cells. So the inhibition of the AKT MMP9 pathway indicating a promising potential target in treating metastatic TNBC. The other one is NF-kappa B signaling pathway. NF-kappa B is an essential transcription factor for regulating apoptosis, cell proliferation, and several pro-inflammatory proteins such as chemokine, cytokine, prostaglandin, nitric oxide, euphratrins, and adhesion molecules. Any dysregulation of this NF-kappa B signaling pathway in the activation of this NF-kappa B enhances cancerous cell survival and contributes to an increment of chemotherapy resistance. In TNBC or triple-negative breast cancer cells, overexpression of this NF-kappa B has been significantly demonstrated in several studies. MicroRNA, especially 146, negatively regulates the expression of NF-kappa B, exhibited an essential role in reducing tumorigenesis and progression by suppressing tumor cell migration and invasion. Flavonoids like quercetin inhibit the activation of NF-kappa B signaling pathway, leading to downregulation of the gene products that decrease apoptosis and enhance tumor cell metastasis. So this is a summary of some selected flavonoids with targeted pathway and also some selected microRNA involved in regulation of this targeting pathway and then finally the main effect. As you can see, flavonoids such as the first row, quercetin, the targeted protein kinase C through the modulation of microRNA 200B, and finally the inhibits. And the rest is the same as how these flavonoids that are very important to target some pathways, which is mostly associated with TNBC and other cancer cells, and also some selected microRNA through modulation of this microRNA, and finally they are used to inhibit the proliferation, metastasis, and progression of TNBC and other cancer cells. In conclusion, microRNA are largely dysregulated in many cancer cells, including triple-negative breast cancer cells, and expressed in most cancers, which has been associated and represent a desirable and potential target for the treatment and prevention of TNBC. So flavonoids display a unique ability to change or to modulate or regulate microRNA levels via different mechanisms, including transcriptional, epigenetic, and microRNA processing, downregulating of oncogenic microRNA, and upregulating of tumor suppressor microRNA. Also, flavonoids can promote chemotherapeutic drug sensitivity to specific cancer cells, indicating a promising potential anti-cancer drug for TNBC. As you can see here in the graph, action of flavonoids through different mechanisms by regulating the pathways and other signal transduction that are used as a potential target site in the treatment of TNBC. Acknowledgement, the fourth virtual conference on cancer science and oncology organizers to invite me to present on this scientific research papers on the scientific conferences and Florida Agricultural and Mechanical University, College of Pharmacy and Pharmaceutical Sciences, and there are these are potential advisors and participants and co-authors of this paper, which are valuable in to be published in these high impact journals. This is the paper that are published and presented here in this conference. And finally, thank you. If you have any question and answer, you are welcome. Thank you.
Yes. Any questions to Dr. Jednet? This was the presentation from Dr. Jednet. If you have any questions regarding this topic, just you can raise your hand. Any? Yes, I think there is no question, sir. Yes, thank you very much, sir, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. Also on behalf of the
thank you so much.
Yes,
thank you so much.
Yes, also on behalf of the
thank you
members of the organizing committee, members of Innova meetings, LLC. I want to thank you for insight presentation. We hope to meet you next time, sir, in physical event if everything goes well in 2022. Looking forward to meet again next time.
Thank you. Okay.
Yes. Thank for having your since yesterday you are staying in this conference, having with patients. Yes. Next, we ask Dr. K. Shija, Division of Cancer Research, Regional Center, Cancer Center, Medical College, Thiruvananthapuram, from India. The topic of the title will be NLRP3 inhibition prevents the innovation and migration of human melanoma cells. Yes, madam, you can start your presentation by sharing your slide. Just to, when you're delivering your talk, just we ask you to have with loud noise, madam. We are unable to hear your voice. Just we are getting very little sound, madam, from your side. Just we ask to give louder sound from your side. Just little bit sound we are getting from your side. Only little bit. We are getting only little bit sound from your side. We need to have very low sound we are getting. No, not yet, madam. Just you can remove your earphones and you can try once if it is possible.
Now it's okay, sir?
It's fine.
Okay. Okay, sir. Very good evening to all, and I'm going to present my research topic. The title of this presentation is NLRP3 inhibition prevents invasion, migration of melanoma cells. And I'm working on the Division of Cancer Research, Regional Cancer Center, Thiruvananthapuram, Kerala. Okay, that is. The tumor microenvironment consists of tumor cells, cellular counterparts, and non-cellular counterparts. Cellular counterparts include stromal fibroblast, endothelial cells, and immune cells. Non-cellular components include extracellular matrix such as collagens. And actually, the dynamic interaction of these cancer cells with the tumor microenvironment determines the cancer cell progression and metastasis and drug resistance. So this intracellular communication is mediated through complex network of cytokines, chemokines, growth factors, inflammatory mediators, and matrix remodeling enzymes. So this picture shows this microenvironment. This is about this, how the microenvironment, what are the components in microenvironment. See, these are the different immune cells and other tumor cells and vasculature. And this interaction which supplies nutrients, amino acid, oxygen, growth factors, and other supporting materials, and also responsible for the cell-cell interactions. So one of the main function of the tumor the mediators of tumor microenvironment is inflammation, combined with the activation of immune cells and the production of pro-inflammatory cytokines. And so coming into the inflammation, which is actually a protective mechanism, but to restore the injury and pathogenic attack. But if it persists for long times, that is unregulated, it may cause a deleterious effect and becomes one of the most important factor which induces the malignant transformation of this cell into a malignancy. So the role of the inflammation in cancer is well documented and described as early in 1863 by Rudolf Virchow. So this inflammation is provide this tumor progression by providing this bioactive molecules from the cell infiltrating the tumor microenvironment, including cytokines and other growth factors, that sustain the proliferative rate. Apart from this, it also enhances cell survival by avoiding apoptosis, providing pro-angiogenic factors and extracellular modifying enzymes such as MMPs, and it also promotes epithelial mesenchymal transition. This information also facilitates other carcinogenic programs such as genome instability, reprogramming of energy metabolism, and immune evasion. Okay, so this is the picture show how this inflammations and regulate the tumor progression. It may induce inflammation by chemokine resistance. Sometimes the inflammation may suppress the immune cells so that the tumor can grow by evading this. Tumor can grow into a higher level. So these are the role of the inflammation and different events of the cancer progression. And this picture shows that this myeloid-derived suppressor T cells can be suppressed by the inflammation so that the cancer can into the progression, leads into the progression to advanced stages. So what are the inflammatory mediators which can be derived from either cells or a plasma? Cell-derived inflammatory mediators, which include histamine, serotonins, and actually, and this nitric oxide and platelet activating factor. These are the factors. Plasma-derived include, this is a different complement factors and this factors 12 of activation. So here, this is important. Cytokines are very important as far as the inflammatory process is concerned. This picture shows how this inflammatory conditions which changes the sum of the immune cell into the tumor associated macrophages, so that can also leads into the progression of the tumor. Here we can see that about of the different factors, cytokines play a very significant role in the tumor progression. So how the cytokines involved in leads to the progression of the tumor. Here comes the importance of pattern recognition receptors. So this pattern recognition receptors are nothing but there are the receptors in the cell which recognize the pathogens or altered cells. They can be of a different type, 4 type, that is toll-like receptors, C-type receptors, not-like receptors, and actually. So this is my interest protein, that is not-like receptors. These are cytosolic pattern recognition receptors. So one of the most, these pattern recognition receptors are node-like receptors are cytosolic, present the cytosol, and some of the node-like receptors can form a special complex structure called inflammasomes. The inflammasomes are nothing but they are a multi-complex protein that are assembled by following the detection of pathogenic or microorganisms or danger signals. It is identified in 2002 and which mediate the stimulation of inflammatory caspases, which leads to the production of large number of inflammatory cytokines, include IL-1 beta and IL-18. So these are the one of the most well studied in inflammosome is node-like receptor protein 3, that is NLRP3, is one of the most characterized inflammosome that belongs to NLR family. So actually this NLRP3 is very important since this can be reactive wide range of inflammatory infections and endogenous signals. Okay, and this actually consists of 3 components: NLRP3 scaffold and apoptosis associated PEC-like proteins and caspase 1. Okay, and these 3 molecules, when a trigger occurs, also it sends a signals, it assembled together to form a complex, NLRP3 complex. So this is a complex. So it consists of 3 molecules. Once this assembled, it leads into the activation. This is a 3rd molecule, effector molecule, which procaspase 1 is present. Upon assembly, this procaspase 1 is cleaved into caspase 1 that leads in the conversion of pro-11 beta and pro-IL-18, which is produced as a part of NF-kappa B signaling into active forms. This overproduction of this IL-1 beta leads into the inflammatory conditions in the tumor site and leads into this progression of the tumor. So here I have studied this influence of this NLRP3 in metastatic melanoma, because melanoma is so important because it is known for its aggressive nature, for its ability to metastasis in variety of atypical location and poor prognostic characters. It is 80% of the death is due to this metastasis of melanoma here, and it in melanoma it is found that upregulation of several pro-inflammatory cytokines. And at the later stage of the disease, autoinflammatory condition is observed due to the spontaneous secretion of active IL-1 beta by constitutive activation of NLRP3 inflammasome. So this NLRP3 inflammasome may contribute toward the advancement of tumor in melanoma. So apart in 2021, it is reported that this NLRP3 in melanoma can also suppress the myeloid-derived suppressor T cells that also leads to the progression of the tumor. So based on this literature, I have studied what is the effect of NLRP3 and tumor human melanoma cells metastasis. Okay, so methodology adopters includes this pharmacological inhibition of NLRP3 using MCC 950, that is inhibitor or specific inhibitor NLRP3, and NLRP3 is activator based using the LPS and lipopolysaccharide and ATP. And these cells, this activator and inhibitor, NLRP3 activator and NLRP3 inhibitor cells were analyzed for the various events of the cancer progression, which includes by colony forming assay and Western blot, enzyme linked immunosorbent assay, and migration and invasion assay, and what are the genetic genes involved in the process of NLRP3 inflammasomes mediated progression of the tumor by using the real-time PCR. So here the observation, this is a result of the Western blot study. While the normal cells shows this expression of NLRP3, by when the trait activate NLRP3 is activated, NLRP the gene is overexpression of the gene, while the inhibition leads into the down regulation of the genes. So this indicates that this chemical treatment we have efficiently down regulate and upregulate the cells. Then these cells were used to study different events. So we will check what is the effect of NLRP3 on proliferation of 375 cell, that is melanoma cell, which is a model cell, which is used for the analysis investigation. So here found that normal cell produce large number of cells, and NLRP3, LPS and ATP NLRP3 activator cells also shows viable cells are more, but in NLRP3 inhibited cells shows less number of viable. This indicated that NLRP3 involved in the proliferation of the cell. So the acridine orange Ethereum bromide staining of A37 cells also detected. This is a normal one, normal in nature, but as active NLRP3 activator cells is also normal. And while this this is a AMCC 950 treated cell, which shows and morphology shows that this is a characteristic of the apoptotic death. So the membrane blabbing, fragmentation of the DNA, etc. This indicates that NLRP3 is involved in this induction of this as apoptosis. Okay, when inhibited, the apoptosis is induced, which leads to inhibition of the tumor. And a colony forming assay shows that normal cells number of colonies are this, and when it's activated, number of colonies are more. That means it induces the progression of the tumor, while the down regulation inhibition of the NLRP3 reduces the number of colonies, and this indicated that NLRP3 is involved in the proliferation of the cell. This is a confirmation test. And here comes to check whether how this is a ELISA. This ELISA result of the ELISA, in which we have quantified the interleukin 1 beta and IL-18 after the inhibition and activation of NLRP3. Here comes the activator cells produce more number of IL-1 beta, while MCC 950 treated, that is NLRP3 inhibitor cell shows less, the level of the IL-1 beta is very less. Similarly, this IL-18, which is another pro-inflammatory cytokine, which is produced as a result of NLRP3 activations, shows reduced level of the IL-18 in the NLRP3 inhibitor cells and high level in the NLRP3 activator cells. This indicated that NLRP3 involved in the production of pro-inflammatory cytokines. So here this is the result of the invasion and migration assay. This is the first one is normal. Normal shows large number of cells, while the inhibited cells shows less number of cells, and while the activation of NLRP3 by LPS and ATP shows more number of cells, which is similar to that of a normal cells. This indicated that NLRP3 activation induces the progression of the tumor, while the inhibition treatment downregulation of NLRP3 reduces the number of the migratory and invasive cells that is invaded to the membrane. This indicates that it is NLRP3 involved enhances the migration and invasion of the tumor cell. Similarly, this is then we analyzed how this activation of NLRP3 and inhibition of NLRP3 affect this gene expression, various genes. That is apoptosis associated spike-like protein complex, so ASC, which is important component of NLRP3, is upregulated on the activation by the downregulator. Similarly, HMGB1, which is about pro-inflammatory cytokines involved in this process, upregulated by NLR, LPS, ATP, and it is downregulated by the inhibition of NLRP. Similarly, NEK7, which is interact with the NLRP3, in order to activate it, is once upregulated on activation of NLRP3, and it is downregulated by EMCC 950. Similarly, NLRP3, IL-1 beta, and caspases 1, which is an important involved in this activation process, found to be upregulated, and it is downregulated. So this means that this NLRP3 inflammasomes upon activation, which induces the progression of the tumor by activating ASC, HMGB1, NEK7, IL-1 beta, and caspase 1. This indicated that this NLRP3 inflammasomes involved in the metastasis of the melanoma cells, other than the cells, by inducing the migration and invasion of the tumor cells. And coming to the summary and conclusion, the result of the present study indicated that NLRP3 inflammasome plays significant role in the invasion, migration, proliferation of cells. This information increases our understanding of the pathogenesis of melanoma, provides clue for developing new attractive preventive and therapeutic approaches by targeting NLRP3 inflammasome in future. And this study was supported by the Department of Science and Technology, Government of India. Thank you very much.
Yes, ma'am. This was the presentation from Dr. K. Shija.
Okay. Thank you very much.
Yeah. Any questions to Dr. Shija?
One aspect, this NLRP3 is actually binding to the ATP. Does it actually release the prevent the access to that ectonucleotide ENTD, CD39? No? Actually, that is one reason where actually cell proliferation and then cell survival pathway is activated.
Okay.
And
Yes,
angiogenesis is activated.
Yes,
in case of diabetes, what is actually happening is that is decreased because this epirise is not there. So,
okay, sir.
So in this particular case, the accessibility of these things, is it prevented in case of cancer? Can it be developed as a specific actually treatment system for cancers? And inhibiting this, does it improve actually vasculogenesis in diabetes? Because these two are opposite things. They are the primary drivers of diabetes on one hand and cancer on the other.
Oh, okay, sir. So actually, this is actually the function of NLRP3 is very controversial, and it is specific to the context. Okay. In melanoma, it is progressing, but in some other cases, it is down-regulating.
Yeah.
So this active action of the NLRP3 is very controversial. It has a dual role. I have studied in melanoma, it is upregulating. So the mechanism is not clear, sir. Why this is controversial? Contradictory role is, it is, okay. So in melanoma, it is progressing. So, and one more thing, sir, as some cases, I also found some contradictory results that I'm going to, as checking about the apoptosis and another is pyroptosis. So we cannot differentiate the pyroptosis and apoptosis in. Sometimes it is activated. Okay.
That is normally associated with the iron sulfur cluster systems. Ferroptosis is aconitases play a critical role in that thing. Actually, that was ignored. Warburg's work, initial work was on that. Actually, he drifted this and destroyed 100 years of research. But the NLRP3, that is a good signature where actually binding by ATP.
Okay.
It is actually denying the access of ATP to apoerase. So that way it prevents angiogenesis.
Okay.
And that is the mechanism, whether actually in case of these intracellular systems, whether in diabetes, this is actually active. This mechanism, if it is active, your vasculogenesis is inhibited.
Okay.
That could be one approach which you need to examine, I believe, in my opinion. That's my opinion, that's all.
Okay, sir. Okay, very well. Thank you, sir, for giving this opinion. One more thing I wanted to ask you, sir.
Yeah.
Sir, do you have any idea about this expression of NLRP3 T cells?
Specific?
T cell, T lymphocytes. T lymphocytes.
No, basically what is actually happening is the partition between differentiation and then proliferation is regulated by that glycosylation process.
Okay.
That glycosylation process, in case of mesenchymal cells, that TGF beta inhibits actually SLC7, that is 4F2HC, CD98 or HC. So that is a co-transporter for essential amino acid uptake. And that is prevented, then your NAD levels will not be properly maintained. Your lipid rafts and then, receptor system at the cell membranes will not be properly maintained.
Okay.
That is one of the major difference between apoptosis or mesenchymal state and then, this thing. It is an adaptive process, as I said earlier, for embryological tissue differentiation. During that particular differentiation, Jyotsna's paper, I believe, she presented a good data on Drosophila, where actually cell migrations will be taking place in post-gastrula stages. So during that period, immunity will inhibit it. So organogenesis actually promotes immunogenesis so that, one tissue will not be interfering with the other tissue function. There will be coordination, but encroaching functions will not be there.
Okay. And it is found that macrophages also release NLRP3.
Yeah, that is macrophage differentiation is because of the accumulation of the glucose in the blood, where actually immune cells, macrophage, monocytes and other things. As I said in my lecture, just take a test tube, put some glucose and then put your bone marrow or other things, you will monocytes will be differentiated into macrophages. That doesn't need any big equipment also. Glucose is a differentiation factor. Glucose is a differentiation factor. It is not an ATP producing factor. It is actually PGK1 and PGK2, these two things. You can also examine this thing in polycystic syndromes in case of ovarian metastatic cancers, whether PGK2 is appearing. In testes, we know clearly.
Yes, ma'am. So you are, yes, madam, you will be having one more question from Dr. Sundaresan. Say about the method used for innovation and migration. You can find from chat box, madam. The question is, say about the method used for innovation and migration.
Okay. Sir, I didn't hear you.
you are getting a question from Dr. Sundarajan. Say about the method used for innovation and the migration.
Invasion. Actually, sir, for the invasion, we are using cells inserts. Cell insert that is containing this membrane with spores. So we are after this particular number of cells, we are suspending in this DME medium, cell culture medium without SBS, and on the upper chamber and the lower chamber, we are adding the medium with chemo in the presence of high concentration of glucose and incubate the CO2 carbon dioxide incubator for 48 hours, and the number of cells that is invaded through the membrane and it is attached the lower side of the membrane is stained and fixed, stained and photographed, and number of colonies, number of cells present on the lower side of the chamber is counted in the microscope. Similarly, we are doing the migration assay, but the difference is that in the migration assay, we are not coating any extracellular matrix or matrix gel on the membrane, but in the invasion, we are coating. We are creating the extracellular matrix, mimicking the extracellular matrix by coating this matrix gel on the surface of the plasma membrane, polycarbonate membrane. That is the procedure we are using.
Yes, ma'am.
I think this can be a good diagnostic feature for identifying diabetic patients, whether this binding of phenolphthalein to ATP is visible in blood circulation of diabetic patients.
Okay. Okay. One more thing I want to ask you, sir. Sir, do you have any idea about this presence of active inflammosome in T lymphocytes?
My audibility is little low.
Yes, madam. One thing, madam. You can ask through chat box, madam.
Can you put your question on chat box? I try to address that thing if I know it. Okay.
Okay, sir. Because I tried in macrophages, but I also wanted to study whether it is present on the T cells. And whether it is affecting the cancer progression or
normally what happens is during the accumulation of the glucose, see, one of the major reasons in diabetes where hyperglycemia is causing is it actually promotes differentiation of macrophages and also immune cells. That is the major reason.
Oh, maybe some it have some influence on the T cells.
That was distorted in 20th century by using glucose as an oxidizing agent. Glucose is a promoter for actually by supplying this thing because that pentose phosphate pathway was actually misinterpreted.
Okay, sir. Thank you very much. Thank you.
It's a good information. It's a new diagnostic feature.
Yes. Thank you very much, madam.
Thank you very much. Okay, thank you.
Thank you very much, madam, for speaking up in this meeting and providing a unique perspective. Thanks for sharing your tips with us. Also, on behalf of members of the organizing committee, members of Innova meetings, I want to thank you for your presentation.
Okay, I'm very much thankful for getting an opportunity to present here and share. I'll get more information from sir regarding the inflammosome. Thank you very much, sir. Thank you
We'll be
for the organizers.
Yes, we'll be sharing the Lakshmipati sir email ID.
Yes, I want. Okay.
Officially, you can through your email ID. Just you can have the conversation.
We can be in touch. We can be in touch on mail also. My mail ID is available there in your.
I want your help, sir. Okay.
Thank you.
Okay,
I'll be
happy.
happy. My only passion is this thing. My only work is this thing. I am socially almost isolated. My family also is not there. Therefore, this is my avocation.
Definitely. Yes. Next we asked Dr. Sabah Abdul Hamad from Sindhu Institute of Urology and Transplantation from Pakistan. The title of the talk will be Role of PET CT in Radiation Oncology Treatment Planning.
Can you hear my voice?
Yes, madam. Go ahead.
A slideshow. Okay, I myself is Sabah Abdul Hamid from working in the Nuclear Medicine and Radiotherapy Department of Sindh Institute of Urology and Transplantation from Pakistan. So I will talk about the role of PET-CT in radiation oncology department, especially in treatment planning. So these are the contents of my presentation. I will discuss about the brief introduction of PET-CT, and then I will represent some research paper, and then I will discuss about the brief introduction of radiotherapy and the PET-CT role in radiotherapy treatment planning, and what are the use of PET-CT limitations. What are the use of what will be the limitations and challenges we will face when we use the PET-CT in treatment planning system. So PET-CT is basically the hybrid PET-CT system, which is the fusion of positron emission tomography with computed tomography. Together, this system provides structural and metabolical information of a cancer cell or any inflammatory cells through detection of 2511 keV photons emitted from patient body after administration of a positron emitting radio tracers. And PET-CT is also provides the early detection and tumor recurrence in early stages than other imaging modalities like computed tomography and magnetic resonance imaging. So this is the PET-CT machine. This machine consists of two compartments. The first compartment is of PET, positron emission tomography, and the second compartment is of computed tomography, from which the patient first undergoes through the computed tomography, from which the scout view of the patient anatomical details are collected as similar to the radiotherapy simulation system. And this is the image of CT scan anatomical details of the patient, and this is the cell activity collected from the PET scan data. And the last images show the fusion of PET-CT system, which also included the anatomical details as well as metabolic activity of the tumor cells or any inflammatory cells. So what are the different radiotracers or positron emitting radioisotopes which are used for the PET-CT scan? These radioisotopes were basically used for staging, diagnosis, and treatment selection, and for therapy to check the therapeutic response in specific patients. So there are several radiotracers were used, but in our institute we use only 3 radiotracers, which are F18 fluorodeoxyglucose and gallium-68 dotatate and gallium-68 PSMA. So F18 is the radioisotope which is labeled with fluorodeoxyglucose, which is the substrate or any ligands in the is the analog of the glucose. Or whereas the dotatate is the somatostatin receptor analog, it is a modified octreotide. Basically, and PSMA is a prostate-specific membrane antigen, which is used for the for detection of prostate-specific antigen in prostate cancer cells. So FDG are used for the metabolical activity of the cancer cells through measurement of standardized uptake value, especially used in head and neck cancer, lung cancer, breast cancer, lymphoma, or melanoma. Whereas the dotatate used for neuroendocrine tumors, which includes the tumors of nervous system or endocrine glands. And the gallium-68 PSMA were used for imaging of prostate cancer. So these are some research paper I included. They discuss about the PET-CT role in treatment planning of radiation oncology. The first research paper is of Johan Bazin et al. They study about the importance of PET-CT in radiotherapy treatment planning and monitoring of their therapeutic response in solid tumors. Solid tumors which includes the head and neck cancer and lung cancer. They discuss about the staging and detection of monitoring of the effects of treatment from different modalities, allowing the oncologists to quantify the resistance mechanism of the cancer cells from after prolonged radiotherapy or chemotherapy system. And also enabling the oncologist to select the patient through proper their through detection of their resistance mechanism, and allowing the oncologist to appropriately select the patient according to appropriate treatment. Whereas the Ramachandran in their study discuss about the insight of PET-CT-based treatment planning and also discuss the significant influence of PET-CT in target delineation of lung cancer as compared to PET or CT system used in alone. So he discussed that to increase the patient comfort, data acquisition time should be collected in less, the duration of data collection in PET-CT scan should be less. And they also discuss about the resolution of PET-CT system with MRI or MRS. MRS, which have the greater resolution of anatomic details than PET-CT. So they discuss about the usefulness of PET-MR system, which will further improve the outcome in radiotherapy in future studies. The last study was done by Frank Sirnek. They studied about the radiation threatening planning with integrated PET-CT system. They basically done a case study in 39 patients with diagnosis of head and neck cancer, lung cancer, and pelvic cancer, respectively. And they do the PET-CT based planning by 2 independent oncologists. Their results showed the significant change in GTV delineation after PET-CT based planning, which is changed in 56% cases, including 33 patients, including the 33 patients from with head and neck cancer, 67% of lung cancer patients, and 90% with cancer of pelvis. There's also revealed that distance metastasis they also revealed this in 60% cases of their patients. The distance metastasis also revealed, which changes the treatment strategy from curative to palliative. So now I will discuss what is radiotherapy. So basically, radiotherapy is an advanced technique for management of benign or malignant tumors with surgery and chemotherapy. And day by day technological advancements in RT like IMRT, 3D CRT, VMAC, and IGRT uses ionizing radiation in a very small field. So precise and accurate target delineation is very essential in treatment planning. So what will be the role of PET-CT in target delineation of or target presentation of a RT planning system? In RT planning, the tumor volume basically defined in 4 tumor volume, which were proposed by ICRU, International Commission of Radiation Units. They proposed GTV, gross tumor volume, which is basically the macroscopic tumor or palpable mass. And then the clinical target volume, which includes the GTV plus microscopic disease surrounding the tumor tissue. And then comes the internal target volume, which is basically the inclusion of clinical target volume with internal margins of 4 organ portions. And finally, the planning target volume is defined as the internal tumor volume plus setup margins or for use for uncertainties which will be occur in during planning during treatment of radiation oncology. And for boost planning, the GTV there will be the significant changes in the GTV delineated, which is delineated from PET-CT based planning. And it is significantly changes from 10 to 50%, reducing the toxicity to normal tissues and controlling the local tumors. And it will providing great target volume definition patients to oncologists by easily differentiating tumor structures from adjacent normal soft tissues or small lymph nodes, thus improving the clinical outcomes. And PET-CT-based treatment planning also has a higher sensitivity in staging of mediastinal lymph nodes and also minimize the motion variability if it is PET-CT scan obtained in the same treatment position as to radiotherapy treatment position. And the PET-CT-based radiation in lung cancer significantly reduces the axis dose to normal lung tissues and heart. So PET-CT-based target volume delineation is an attractive alternative for radiotherapy. And this is the image of a patient of head and neck squamous cell carcinoma. The PET-CT scan of this patient is done in the treatment position or as similar to radiotherapy, and the patient will immobilized with thermoplastic mask. So in the first image, A represented the CT image of the patient and the B represent the PET image and C represent the fusion of PET-CT. So the planning was done on the fusion of PET-CT image, and the GT, this is the red outline shows us the gross tumor volume of a, and the surrounding of a GTV is a clinical target volume, is the small red boundary, and the blue boundary shows us the planning target volume in which the setup margins are selected for any motion uncertainties. And the green and aqua regions are of normal elective nodal volumes for contouring of organ at risk in order to minimize the dose to organ at risk. So what will be the limitations if we use the PET-CT system based in the treatment planning system of radiation oncology? The major limitations will be is that the PET tracers are not specific for tumor imaging. As I first discussed, that it will also provide us the image of inflammatory cells from metabolic in which metabolic activity is very high. So the chances of false positive results can occur in inflammatory cells. And PET images are of lower resolution as compared to CT or MRI. As since it cannot define the anatomical structure in very clearly, so it will be co-registered with the CT or MRI for to getting the better clinical outcomes. And the PET alone cannot use for RT dose calculation, as I discussed above, that it cannot provide us the anatomical details of the patient. So we proper cannot define the adjacent structures or peripheries of tumor in using the PET alone. And PET has a very long acquisition time. So during the FDG PET system, which is the analog of a glucose. In this protocol, during the FDG PET scan protocol, we ask the patient to relax for 45 to 60 minutes and don't use any muscular activity, so avoiding the glucose level in normal tissues. So that's why it has a very long acquisition time, increasing the patient discomfort. And finally, the patient positioning tool should be similar as used in simulation or linear accelerator of the radiation oncology. And then PET-CT console softwares must be able to provide radiation therapy structure set data in digital imaging of communication in medicine. The PET-CT contouring system or software should be linked with a radiotherapy platform system, as it similarly provides the contouring mechanism as it is represented in RT planning system. And in cases of progressive malignancies like in normal small cell lung cancer and head and neck cancers, PET-CT images should be used as a descent, because these cells are progressively proliferating, which could changes the microenvironment of cancer cells very progressive. So in this case, capacity images should be used in recent for RT planning. And what will be the challenges if we use capacity-based radiotherapy planning? The first will be that the automatic GTV delineation from capacity data faces big challenges due to tumor heterogeneity and stability of pressure uptake. Tumor heterogeneity changes basically then the mobility and proliferation are continuously occurring in cancer cells. So different heterogeneity will be represented in GTV. If we use the PET-CT metabolic system, it will very limited to our GTV. And the main challenges for PET-CT is that in cases of lung cancer, respiratory due to continuous respiratory motion of a patient, the image noises will be so high. So for during contouring, the image reconstructing software should be used by applying the motion estimation and correction. Thank you. Any questions?
Yes. This was a presentation from Dr. Sabah Abdul Hamad.
One question, please.
Yes, yes, sir, go ahead.
Say, I just want a clarity on this thing. GTV, what do you mean by GTV? I missed your voice actually, and also missed a few slides. Yeah, what do you mean by GTV?
Okay, I will repeat it.
I'm not able to hear you. Hello? Can the coordinator help me?
Now will my eyes is clear? Voice is clear?
Yeah. I am specifically interested in one question, that is 2-deoxyglucose, which is considered to be actually AMPK product. Always we think about what is called AMPK whenever it comes, we link it to LKB and other things. But AMPK, if I am not wrong, also activates 2-deoxyglucose in case of this thing, that reactivates actually insulin signaling pathway. That is one mechanism. How does actually these two scans arrest or enhance the 2-deoxyglucose within the cells?
I will not get your question.
2-deoxy, what will be the position after the scanning about the 2-deoxyglucose position within the cells? 2-deoxyglucose hampers actually glucose uptake, but activates actually the IRS signaling, IGF-IRS, insulin receptor substrate signaling.
Yes. So the glucose uptake occurs that in kinase pathway is
not able to hear.
No, not here, able to hear, madam.
You are not able to hear.
Okay. Will you get
I want
my
clarity
voice?
on two things. I want clarity on two things. I don't know whether you are able to hear me or not. Number one is, what do you mean by, how do you explain GTV? That is number one. Number two is, what will be the position of 2-deoxyglucose between the two scans and combined scan?
Okay. So speaking honestly, I will don't know about the second question. The position of deoxyglucose in combined modalities. I will research about it and get in touch with you through email. Okay, is that okay?
Yeah, you can just mail me the reply in case you, or you can put in the chat the reply.
Yes,
I'll be in a position to actually understand that. Thank you.
Yeah, I have it. And very
Thank you
sorry
so
for
much.
and GTV. The GTV, gross tumor volume, is basically the palpable or visible mass which is visible during the
Audibility is not there.
audibility.
Okay.
Okay.
Yeah, I think sir is not here, madam. Just you
It's
can.
okay.
Yes.
It's okay. It's okay.
Yes, one more question, madam, from Dr. Sundarazan. Thanks for lucid presentation. And I need your PPT for my students will make. Okay, this I'll be sharing, sir. No need to worry.
Okay.
Yeah,
Okay.
I will share it.
Yes. So this was a presentation from Dr. Sabha. Yes. Thank you very much, madam, for joining this event.
Thank you for
Yes.
inviting me.
Yes. On behalf of organizing members of Virtual Cancer Summit 2021, we want thank you for your presentation. Looking forward to meet again next time.
Bye.
Yes. Next, we asked Dr. Napi Falamonkolenka from Central University of Technology, Free State from South Africa. The title of the talk will be the lived experiences following radiotherapy for prostate cancer in Johannesburg, a qualitative study. So, are you ready with your PPT, sir? Dr. Nape.
Hello, how are you doing?
Yes, sir. Yes, sir.
Can you hear me?
It's going to start. Yes, I can hear your voice.
Okay. Let me project my presentation, okay?
Yes, sir.
Yes.
Go ahead, sir.
Okay, all right, that's good. I believe you can see me that side.
Yes, sir. It's fine.
Okay, can you see my slides as well?
Yes, sir.
Okay, all right, good. Let me just minimize this. Okay, my title of my presentation is The Lived Experiences Following Radiotherapy. Are you moving my slides or
no?
Am I the whole?
We don't have any rights to. We don't have any rights to change your slide. It is your.
Okay. Okay, all right. That's good. The title of my presentation is The Lived Experiences Following Radiotherapy for Prostate Cancer in Johannesburg, a qualitative study. I've got no conflict of interest for me to declare in this presentation. For the background, the erectile dysfunction is prevalent in patients diagnosed with prostate cancer undergoing radiotherapy. The literature reports psychosocial distress experienced by these patients following diagnosis and treatment of prostate cancer, causing a decline in sexual dysfunction and psychosocial well-being. This study aimed to explore and describe the lived experiences regarding sexual health for men diagnosed with prostate cancer following radiotherapy. Oncology departments worldwide lack in providing sexual health care, despite ED prevalence in patients diagnosed with prostate cancer. Sexual health education is seldom integrated into teaching, curricula, and work-integrated learning in the hospitals for students training to become radiation therapists or oncology nurses. Talking about sexuality issues is regarded as a taboo in many cultures and religions. A qualitative method of data collection with a phenomenological study design was followed in this research. One-on-one interviews were conducted to collect qualitative data. Study population comprised of 305 patients treated annually at the research setting. A purposive sampling was utilized to recruit information-rich patients treated between 2011 and 2013. Data for this study was conducted in 2015. Data saturation was achieved subsequent to completion of nine one-on-one interviews. Interviews were transcribed verbatim, analyzed using text method of qualitative analysis. Inductive logic of reasoning was adopted in drawing up the themes from the qualitative data. So the results that we developed from the qualitative data is that we found that the sexual experience of the patients after a prostate cancer diagnosis and radiotherapy. So there was some sexual experiences of the patient after being diagnosed and being treated with radiotherapy. So the other theme talked about the impact of losing sexual function on relationships. Theme number 3 talked about the lack of information regarding sexual health among oncology healthcare professionals. So these are the sub-themes that we'll discuss under each theme. So under theme number 1, the sub-theme talks about the physical, mental, and emotional distress. So these are the quotes from the patients, from the data that talks to this sub-theme. For an example here, a 59-year-old patient indicated that just can't get an erection. Sometimes it will come 30%, 45%. So this gentleman was referring to his erectile function. And then a 52-year-old male, I quote, "The last time I had satisfying sex, it was somehow very long time ago. Let me say sexual performance to satisfy my woman," close quote. So this was from a 52-year-old male. So when we move to sub-theme number 2, which talks about the sexual and the emotional detachment feelings, I'll also quote the quotes underneath this sub-theme. The first one, it was from a 59-year-old male. I quote, "She's supportive, but sometimes she gets moody. Then she will pass remarks like, 'I hope tonight she will gonna be lucky,' something like that there," close quote. Let's move to theme number, this other one here. I'm just moving something that is disturbing me. It was a 59-year-old male. I quote, "I even bought her this electrical thing. I couldn't just deny her to have her own desire and all that," close quote. So let's move now to sub-theme number 3. Just to remind you, sub-theme number 3 talks about the lack of information regarding sexual health among oncology healthcare professionals. So now I've realized that there was a lack of consistency with sexual health care. For an example here, a 62-year-old male indicated that, I quote, I said to her, "I got problem," and she said to me, "Okay, she will make a note of it when I come again." So this was a doctor talking to the patient. Another quote here from a 68-year male, I quote, "You see, I pick up a problem with this, as you know, every time you see a different doctor." So this was a patient explaining to me that there's a problem every time I see a different doctor, and it becomes so difficult to follow up on your problem. So that was a discussion that I was having with the patient when we were collecting the data. So I decided that, okay, we need to structure the sexual health counseling process in the radiation oncology department. So the first thing that I realized that needs to happen is that there must be a patient-centered decision-making. So the patient has to be part of the decision-making and has to be at the center. Everybody has to work around with the patient. So now, for an example, if we move to stage number 3, which talks about the pre-treatment counseling, the patient needs to be counseled about the treatment options which are available for him to consider. Either it can be surgery, hormonal treatment, or radiotherapy. So the patient has to, consider which one will be good for him. And also depending on the doctors, of course, they will provide an expert advice on the treatment that is suitable for that particular patient. As you know, that sometimes they don't need to give you treatment, they'll just have to watch you for some time. So the doctors, of course, they will provide an expert advice, but they need to give the patient options when necessary so that the patient can make a choice. And thereafter, administer the treatment of choice. And then when we move to post-treatment counseling, there has to be a communication between the patient and the doctor. And here we can bring the spouse if possible. The spouse can come in so that the patient spouse understand what her husband is going through, so that it's not a problem at home. Because in some cases, I discovered that there was some, divorce going on, lots of problems. The spouse is not trusting the husband, is thinking the husband is going elsewhere when he's going for the treatment. So that is needed. It's very important. The couple counseling is needed. It can even happen at this stage of pre-treatment counseling. So also post-treatment counseling, so that the spouse is aware of what the husband is going through. So the very important part also here is the treatment follow-up visits, because sometimes you will know that the radiotherapy treatment or cancer treatment, there will be somewhere in the big city, at the big hospital, and sometimes patients are from the rural areas. Sometimes patients don't follow up very well with their doctors. So that is very important that the patients also come for their follow-ups when it is the right time for them to come to the hospital. So also I had to sit down and think about the guidelines to promote sexual health care in radiation oncology. For an example here, I had to think about how to initiate sexual health awareness to change the attitude of oncology healthcare professionals, because they've got different views about it, and how do we change their attitude so that it become a norm, so that it can be included in their care that they provide to patients, so that it's not really a taboo to just engage about it, because we know that once you've got the prostate cancer and you're undergoing hormonal treatment, the sexual function will deteriorate. That one is given, we know that. So, and also review the approach of oncology healthcare professionals to sexual healthcare, how they tackle this issue of sexual healthcare in the department, because some of them, they are doing it. We need to know how are they doing it, what informed what they are doing. It is just something that they do out of love, or maybe, we need to know what kind of approach are they using, so that we can have a streamlined approach on how we go about sexual health care in radiation oncology and even in other oncology departments. And also here, we need to consider sexual health as part of managing patients with prostate cancer, because that is a very important subject for men. You will see them when you treat them in a radiation oncology department. They will be sitting somewhere at the corner there talking about their challenges, some of them lying about their situation, that they have the erectile function. Some, sometimes men, they don't really, talk about these issues. So someone to say that is weak is a sign of, a weakness, or, so it is a challenge. So, but if we can make sexual health as part of managing patients, I think also the patient, they will understand very well that they are in this situation together. And also include sexual health into oncology education and training curriculum. so that when you are training, when I was training as a radiation therapist, it was not really a subject that was included. I'm not sure if currently it is well integrated into the education and training curricula of the trainees. So it's something that needs to be integrated so that it becomes something normal for people to talk about to patients so that they feel comfortable and equipped. So here, as you can see, also here I talk about develop a capacity of trained oncology healthcare professional to facilitate sexual health care. Okay, so if you develop that capacity, you will have people that are, are ready to just talk about this and include it into their care, especially when you are counseling the patient for the first time coming into oncology, because you need to talk about the effects of radiation, how the acute effects, the long-term effects. So that can also be part of that discussion there, so that it's not just something that you talk about it as an isolated subject. It can be very difficult to just talk about it as an isolated subject. Establish men's sexual health clinic in radiation oncology. If there is a men's clinic in that radiation oncology, it will be something that, if there's a women's clinic, men's clinic, no one will really, think about, if you go to a sexual health clinic, it means you've got a problem. But if there's a women's clinic, a men's clinic, it will be something that is acceptable. Of course, even women, they have their own challenges when they undergo a cervical treatment. They go issues like vaginal, stenosis, where it's get narrowed and so on, and it becomes issue. There are issues when they engage in sexual intercourse. So these are the things that needs to be incorporated in the radiation oncology, so that it is not just about curing the cancer with radiation and becoming so disease orientated. We also need to be orientated about a human being holistically, about what they do outside the radiation oncology department. And also disseminate educational information on sexual health in multiple languages. So that can be a very important issue, because you will treat some patients coming from other regions and you find that they don't understand the language that is spoken in that particular region. So information has to be available in different languages. And also address doctor-patient communication challenges in the clinic, because some doctors are not really very good communicators. So that can also be a challenge. You do find doctors that are good communicators, and given the fact that in oncology, it is a very a busy clinic, very busy clinic. So a long conversation with the patient also can be a challenge to a doctor, because they need to see other patients. So, but this can also be addressed on how can we improve communication with patients. The other thing is that we need to involve the cancer support groups in patient cancer treatment. So there are patients that have completed treatment that are always available to talk to patients and share their experience of what they had while they were receiving treatment, so that they can inform these men on how they handle their household issues with regard to sexual health issues. So it will be much more easier if another man tells you this problem on how he handled the situation at home, other than just giving, somebody telling you and he has never experienced that problem, even though he might be a healthcare professional, but he has never been through that kind of a problem. And also age, it can be a problem, but if you've got men of the same age advising you about this problem, because he is a living experience of that kind of a situation. So that can really be useful, and the departments needs to really support the cancer support groups that are existing in the communities, and recognize them and make sure that they are part of their cancer treatment plan. So in conclusion here, I just wanted to say that the loss of function, the loss of sexual function affected men's quality of life, psychological well-being, intimate relationship. Oncology clinic prioritize the management of the disease with little emphasis on sexual health issues. And the proposed guidelines for sexual health care are anticipated to improve male sexual health care in the radiation oncology department. So these are some of my references here with some of an article that is published on this work. And I thank you very much. I was short, but I think I just wanted to go straight to the point. That was my submission today. Thank you very much. Program Director, thank you.
Yes, sir.
Yeah, well, it's a very good proposal in terms of advisory role to the medical as well as political administration of the countries in the East. But one paradoxical situation here is, I know that, both cancer and also diabetes are neurometabolic disorders. One of the major difference between vasculogenesis and this thing is, you have actually this phosphodiesterase function. I think Viagra and Cialis and other things act as phosphodiesterase in cutting short cyclic AMP pathway. That is, cyclic AMP pathway cutting down is an important factor between angiogenesis and suppression of vascularization. I address that particular problem. Now, the question here arises is the paradoxically opposite issues, both diabetes as well as carcinogenesis, are affecting the sexual health. In case of carcinogenesis, is it the physical blockade of the vascular vessel that leads to this particular thing, or is there any other way where actually the blockade of blood into the your penile organ is inhibited?
You understand?
Yeah, thank you. As you have seen my presentation, I tried to present it more on a surface level, not going to the biology of what causes the erectile dysfunction. So, of course, when the patient starts treatment, the doctor will have to investigate what, because also the surgery itself, it can cause the erectile dysfunction, depending on how it was done. So the patient needs to be informed about what you are talking about, sort of, because in most cases, they are being told not to take your Viagra when they are receiving treatment, because they are suppressing the hormones during treatment when they are treating the prostate. So you don't want,
Is there a difference in the effect of Viagra between these two, diabetes and cancer patients, prostate cancer patients?
I say is there a difference?
They differ. What is the effect? What is the effect between the two patients, diabetes and prostate cancer patients?
There isn't much of a difference because in terms of the erectile dysfunction, it eventually becomes the same, because in my study, I didn't really focus much on patients which had diabetes, but there was a patient who had a diabetes that I asked, how is your erectile function? And he indicated that I don't have it at all. This started when I had diabetes. So, and then I started to conclude that even though you had the erectile dysfunction before the treatment, so the treatment cannot be the cause of your erectile function. It will be related to diabetes. So in a nutshell, these two cohorts, if I were to study them, I don't think I will find any difference in terms of erectile function.
Yeah.
Because some they will have to, I know some diabetes patients, they live on on injections from men's clinic that they receive. They will be getting tablets like CLS, depending on their severity of their diabetes as well.
Thank you.
Thank you very much. Yeah?
Yes. This was a presentation from Dr. Napi.
No, anyway, I'm not yet a doctor. I'm still working on my PhD.
Yes, all the best, sir. All the best.
Okay, all right. Thank you very much.
Yes. Thank you very much, sir, for speaking up in this meeting and providing a Thanks for sharing your tips with us. Also on behalf of organizing committee members of Virtual Cancer Summit 2021, we want to thank you for your instant presentation. Looking forward to meet again next time in physical event in 2022 if everything goes well.
Thank you very much.
Yes, definitely, sir.
Bye.
So, if you go according to the program, we do have more 3 speakers are left for the conference. That is Dr. Dario from Netherlands, Dr. Daniela from Argentina, Dr. Il Adji from France. So, just we'll wait for 10 minutes break, and just let's see if remaining 2 speakers are going to join their this session or not, or else we'll end the day 2 of the conference. So just we'll have 10 minutes break, because just we are running 30 to 40 minutes ahead of actual program, so we need to manage, because one has to join from Paris, one has to join from Argentina, one has to join from from the Netherlands. So just we'll wait for 10 minutes, and we are meanwhile we'll contact them, and or else we'll end the day 2 of the conference. If you have any queries or questions, just you can have B2B meeting. If you're interested now, just you can have for 15 minutes, 10 to 15 minutes, any B2B meeting. Sir, just we are going to close the program, sir, for today's second day of the conference. Yes, thank you very much, sir. A small testimonial from your side. It was a small testimonial from your side. Yes, if you go to word of thanks, thank you very much to all the participants who had joined this webinar, having different time zones. Our most sincere thanks to all the keynote speakers, plenary speakers, students, delegate, journal collaborators, media partners who have joined from different destinations around the globe for their support and cooperation. Let us thank speakers who have responded so well to our invitations to participate in this conference. We have received several papers which will form the basis of our discussion in the various sessions. Let us thank you all associating with this conference by your presence. And once more, a special thanks to Program Manager, Mr. Henry, Ms. Nadia, for his and very continuous effort putting us together under one platform. If we go to the day two keynote speakers, Dr. M. Constantine Saman, Dr. Lakshmipati yes, speakers Mr. Napi, Dr. Jetnet, Dr. Shija, Mr. Mahmoud Jabair al-Islam, Ms. Sabah Abdul Hamad, Dr. Pavitra Balakishan, Ms. Shanaf Sharif, Dr. Neeraja, Dr. Anna Fastino, Dr. Muhammad Muzdaba. These are the participants who delivered their talk today's sessions. And also, if everything goes well in 22, hope we can join in physical event on same topic will be that Cancer Science 2022. Actually, we are planning either in Dubai or Singapore or Europe, just like best cities like in the world, either Italy or Spain or France. And a special thankful to again Dr. Lakshmipati as well as Dr.
Thank you very much.
Yes, yes.
Thank you very
yes.
much for waiting and also inviting me.
Yes, Doctor.
I wish my health permits to participate in 22 also. I love this thing. I love only this thing. I don't have any other social activities as such. So thank you very much. Anybody who has participated in this want to interact also, I have few questions to be addressed. I'll be happy in the areas also to. I don't have any working lab as such.
Yes,
Internet is the only one. I spend my own money and time on that. Okay, thank you very much.
Yes,
thank
sir.
you very much for that.
Yes. I would also like to thank participants who we can collaborate in the future events, either in physical or virtual events. And one more thing, kindly note, the electronic booklet as well as electronic certificate will be sent through email within 3 to 4 working days after the completion of the conference. And finally, we extend our appreciation to each of you for your participation in this online conference having this pandemic period. So, shall we close the program? So we are going to close the program within 1 or 2 minutes. Yes. Thank you again Have a nice day. Bye. This is signing off. Bhupati.